considered a safety parameter in the trials, it is discussed here given its close association with insulin effectiveness. We'll start with type 1 diabetes. Before presenting the detailed design of Trial 171, I'd like to highlight some key points. The trial was designed with input from the agency to assess the efficacy of TI using the new Gen 2 inhaler for type 2 diabetes in a non-inferiority design. The comparator is a standard of care injected prandial insulin, insulin aspart. A switch trial design was chosen. All subjects were required to convert to insulin aspart prior to randomization. This occurred at the beginning of a 4-week pre-randomization period when basal was being optimized. The patients who were subsequently randomized to TI needed to switch to the inhaled insulin while the aspart arm continued on injectable aspart. Importantly, there was not a separate dedicated basal optimization phase for subjects randomized to TI. Let's now look at how these design elements played into the detailed design schematic. The study was broken down into 4 key segments. During the initial 4-week basal optimization phase, patients were converted to prandial insulin aspart, and basal insulin was titrated every 3 days. If subjects responded to aspart and had an FPG less than or equal to 180 mg per deciliter at the end of this phase, they proceeded to be randomized to one of the 3 arms. In the active comparator arm, subjects remained on insulin aspart plus basal insulin. In a second arm, subjects switched from aspart to TI using the Gen 2 inhaler. During the 12-week titration period, subjects titrated their TI dose based on 90-minute post-meal glucose values. Titration in either arm occurred up to once weekly. As agreed with the agency, a third arm was included for a safety comparison between the 2 inhalers. Following the titration phase, the next 12 weeks of the randomized treatment phase was to be stable dosing except for safety reasons. The primary efficacy comparison was between the TI Gen 2 and aspart arms. During the 4-week follow-up phase, all subjects returned to aspart. The non-inferiority margin was 0.4%, which is the standard non-inferiority margin for insulins. The population was the full analysis set, which is comprised of all randomized subjects. The primary analysis method was mixed model repeated measures, or MMRM. The demographics of the TI and comparator groups in Trial 171 were generally well matched. 40% of the subjects were from the U.S. The subjects were on average overweight. In Trial 171, there were more dropouts in the TI Gen 2 arm, 25 versus 11%. 16 subjects in the TI Gen 2 arm discontinued due to adverse events, mostly cough and other pulmonary adverse events. In addition, 17 subjects in this arm withdrew consent. These were frequently for personal reasons. 5 subjects dropped out for perceived lack of efficacy or concerns about hyperglycemia. 8 events of 19 in the aspart arm were for withdrawal of consent, all of which were for personal reasons. The primary analysis demonstrates TI's efficacy in type 1 diabetes. The A1Cs at baseline were both about 7.9%. The decreases from baseline to week 24 were 0.21 and 0.4. The between treatment difference was 0.19, shown on the right, with the upper bound of the 95% confidence interval being 0.36, satisfying the non-inferiority criterion. Some additional points. MMRM, the primary analysis method, is the preferred approach for handling missing data in longitudinal clinical trials. Assessment using the per-protocol population also supported non-inferiority. The impact of missing data was evaluated by multiple sensitivity analyses. Non-inferiority was concluded in all the analyses except the one using the worst case imputation against TI. In the Trial 171 time course, A1C levels were comparable for both treatment groups at screening around 8.5% and baseline 7.9%. A1C declined in the first 12 weeks in both treatment groups, more in the aspart group, and then remained stable during the stable dosing period. Thus, TI maintained glycemic control in this population with type 1 diabetes over 24 weeks. Let's now look at basal and prandial dosing data for the two study arms. This time course shows the basal dose over the 24-week randomized treatment phase. The doses increase in both arms during the initial 12-week titration period and are stable during the subsequent stable dosing phase. The increase in the mean daily basal dose was approximately 3 units more for TI versus aspart subjects, consistent with TI's ultra-rapid kinetics. An analysis of the primary efficacy endpoint that corrected for the differences in basal dose did not affect the efficacy conclusions. The prandial aspart dose started at 23 units at randomization, labeled baseline or BL, with net change in dose that is small. This is not unexpected, given that all patients had used aspart during the basal optimization phase, and about 40% of the subjects entered the trial using aspart. However, despite the small change, there was a mean of 11 titrations per subject in the aspart arm, approximately equal numbers of dose increases and decreases, indicating subjects in this arm were continually adjusting their dose. The doses of inhaled TI are expressed in TI Gen 2 cartridge units. At baseline, the mean daily dose of TI Gen 2 was 75 units. The baseline TI dose then increased by 35 TI units. The dose remained relatively steady during the stable dosing period. Trial 009 was the previous 52-week supportive trial of TI in subjects with type 1 diabetes. It was a non-inferiority comparison of TI using the Medtone inhaler versus insulin aspart, both in combination with basal insulin. The primary endpoint analysis of Trial 009 is shown here. The upper bound of the 95% confidence interval of the treatment difference of A1C change is at 0.404 rather than being less than 0.4, so strictly speaking, non-inferiority was not satisfied. The significance level of the non-inferiority is 0.056. The curve shows that either TI or insulin aspart in combination with basal insulin maintains A1C steadily over the 52-week trial. In order to put the effect sizes in our two active comparator type 1 trials in perspective, this table provides A1C data from the 6-month registration trials of the last approved prandial insulins. The fourth column shows mean changes in A1C from baseline to final study week. The changes from baseline to 6 months are similar to those of Trial 171, which is shown at the bottom of the table. The secondary endpoint data will now be presented for the two type 1 trials, Trials 171 and 009 together. These endpoints provide important context for understanding the clinical utility of TI. For the secondary endpoint data, p-values are provided for descriptive purposes only. This is because the approach for protecting type 1 error was either not fully specified or because an early stage in a step-down procedure was not passed. For the secondary endpoint of the percent of subjects achieving A1C less than or equal to 7%, in Study 171, aspart shows 30% versus about 18% for TI. In Study 009, the rates are the same in both arms. These are 7-point glucose profiles for Trial 171 at baseline and week 24. They're based on self-monitored blood glucose, or SMBG, data. At baseline, the same pattern of postprandial glucose changes over the course of the day is seen in both arms. At 24 weeks, the aspart follows the same pattern of glucose changes as seen at baseline. The TI arm, however, shows some blunting of the postprandial glucose changes. There is a weight advantage for TI compared to insulin aspart in Trial 171 as well as Trial 009. In Trial 171, a benefit of 1.3 kilograms was seen in favor of TI. This was seen even though the data analysis corrected for differences in A1C attainment. Moving on to type 2 diabetes. Trial 175, which used the Gen 2 inhaler, was a 24-week, double-blind, placebo-controlled add-on trial. This trial enrolled insulin-naive subjects with type 2 diabetes and inadequate glycemic control on metformin or 2 or more oral anti-diabetic drugs. A1C had to be 7.5 to 10% at screening. After a pretreatment phase in which the subjects continued to use their previous oral anti-diabetic drug, or OAD, regimen, the subjects were randomized to add on TI Gen 2 or inhaled placebo, Technosphere powder, or TP. There is no basal insulin. In the 6-week pre-treatment phase, subjects received diet and exercise counseling and were taught SMBG procedures. At randomization, all subjects started on a single 10-unit cartridge per meal of inhaled study drug, TI or TP. During the first 12 weeks of the 24-week randomized treatment period, dosing was adjusted based on 90-minute postprandial glucose or PPG values. Adjustments could be made once per week. Rescue therapy was available if necessary. Over the next 12 weeks, dosing was stable except for safety reasons. After the 24-week treatment phase, all subjects returned to usual care. Trial 175 was a superiority trial. The population included all randomized patients, the full analysis set, and the primary analysis method was MMRM. The demographics were well matched in Trial 175. 1/3 of the subjects were either non-Hispanic Black or Hispanic. Half the subjects were from the U.S., and the population was, on average, obese. The dropout rate was 15% in the TI Gen 2 arm and 21% in the TP arm. Dropouts for AEs were mainly cough or other respiratory AEs in both arms. Dropouts for withdrawal of consent were balanced. In this analysis of the primary endpoint for Trial 175, the baseline A1C value is 8.25% in both arms, decreasing by 0.8 in the TI arm and 0.4 in the placebo arm. Superiority of TI relative to placebo, p-value shown, demonstrates the efficacy of TI as add-on therapy in type 2 diabetes. The impact of missing data was evaluated by multiple sensitivity analyses, and the primary findings were confirmed. This graph shows A1C levels over time for Study 175. There were similar decreases in A1C level in both arms during the 6-week run-in. The curves diverge at randomization, with the TI arm dropping more than the placebo arm. Significant glycemic improvement can be seen in the TI arm as early as week 2, while the differences versus placebo continue to increase through week 12, then stabilize during the 12-week stable dosing period. Trial 102 was an active, controlled trial assessing the subjects with type 2 diabetes poorly controlled on insulin with or without OADs. The trial was a non-inferiority comparison of subjects randomized to TI with the Medtone inhaler plus basal insulin compared to a well-recognized basal prandial premixed combination product, BPR 7030, which was administered twice daily for 52 weeks. Looking at the right side of the table, the primary A1C efficacy endpoint for trial 102, the upper bound of the 95% confidence interval, 0.29, with significance level of 0.001, indicates the non-inferiority of TI using the Medtone inhaler plus basal insulin versus twice daily BPR 7030. The time course shows that the two curves are virtually superimposable over the 52-week trial, indicating the comparability of the two treatments and the durability of the A1C response. Turning now to the secondary endpoints for the type 2 trials, 175 and 102. As in the type 1 trials, p-values for secondary endpoints are provided for descriptive purposes only. For study 175 on the left, nearly 40% of the subjects in the TI arm achieved A1C less than or equal to 7% at week 24. The 7-point glucose data for trial 175 shows a clear TI effect on postprandial glucose levels in type 2 subjects. Compared to baseline, at week 24, we see near flattening of post-meal glucose changes in the TI arm. The TI pattern reverted to the baseline pattern at 28 weeks when the subjects were taken off TI. This is included in the sponsor briefing book. The data from the subset of subjects in trial 175 who titrated their breakfast TI dose up to 60 units or greater provided an opportunity to assess the within-subject dose response for the reduction of post-meal glucose excursions. As they titrated their dose up, their post-meal glucose increases diminished. These data show that a dose response occurs for TI from 40 to greater than 60 units. A similar pattern is seen for lunch and dinner. In trial 175, a modest weight gain was noted for TI subjects, about 1/2 kilogram. In study 102, there is less weight gain, approximately 1.6 kilograms less, with TI plus basal when compared to active comparator, consistent with the results seen in the two active controlled type 1 trials. Next, hypoglycemia data for type 1 and 2 diabetes combined will be presented. This slide describes the methods of data collection and the definitions of hypoglycemia for the 4 trials. The definitions evolved consistent with the professional guidances that were in place at the time the trials were conducted. Severe hypoglycemia in trials 171 and 175 meant the assistance of another person was required, not just requested. This table summarizes the incidence and event rates for the active controlled trials. Starting with total hypoglycemia in trial 171, the event rate per 100 subject months shown on the right is 30% lower in the TI arm than the comparator arm. For severe hypoglycemia, both the incidence and event rates are lower in the TI arm. The event rate for severe hypoglycemia in the TI arm is about 43% lower than that of the comparator group. In trial 009, the incidence of total hypoglycemia is significantly less in the TI arm. In trial 102, the hypoglycemia incidence and event rates are lower in the TI arm than the comparator arm. The event rate for severe hypoglycemia is 68% less for TI plus basal than the twice-daily premix comparator. In the placebo-controlled type 2 trial, trial 175, which is not shown here, TI use was associated with more hypoglycemia, which would be expected in a placebo-controlled trial. This is discussed in the sponsor briefing book. Let's now turn to several analyses which show that the reduction in hypoglycemia incidence was not simply the result of reduced A1C reduction. This series of bar graphs compares the hypoglycemia rate of the TI versus aspart arms for 5 different subsets of subjects based on the ranges of A1C levels at week 24. For all of the A1C ranges, which are shown on the x-axis, the hypoglycemia event rate in the gray insulin arm is greater than that of the blue TI Gen 2 arm. The top row of this table shows the total hypoglycemia event rates in trial 171 expressed as model-based estimates. In the second row, the model is further adjusted for A1C levels. Even with this adjustment for A1C, the total hypoglycemia event rate is 27% lower in the TI Gen 2 arm than the aspart arm. The corresponding reduction for severe hypoglycemia using this model is 43%. This slide provides biological plausibility for TI's hypoglycemia advantage, namely that it is due to TI's ultra-rapid time-action profile. In the pair of bars for weeks 1 to 4 of trial 171, the event rates for hypoglycemia occurring from 0 to 2 hours after a meal are shown in the lower portions of the 2 bars for TI Gen 2 and aspart. They are roughly comparable, 2.8 and 3.9. The lighter colored top parts of the bars show the rates for hypoglycemia occurring greater than 2 to 5 hours after the meal. The rate is more than 3 times as high for aspart than for TI, 5.9 versus 1.8. The same pattern of roughly comparable rates at 0 to 2 hours and much higher rates for aspart at greater than 2 to 5 hours is seen at all time periods during the randomized treatment phase. During follow-up weeks 25 to 28, when all subjects go back on prandial aspart, even those labeled TI Gen 2, because that's their original assignment, the differential at greater than 2 to 5 hours disappears. These results underscore a clinical benefit of TI's ultra-rapid kinetic profile. The next 2 slides summarize efficacy. The FDA recommended that we conduct 1 trial in type 1 diabetes and 1 trial in type 2 diabetes to assess the efficacy of the Gen 2 inhaler. Both trials met their primary endpoints with highly significant p-values. The supportive trial for type 1 diabetes showed a trend toward statistical significance. The supportive trial for type 2 diabetes met its primary efficacy endpoint and was clearly statistically significant. In conclusion, trial 171 in type 1 diabetes showed non-inferior A1C reduction of TI Gen 2 compared to the insulin aspart comparator. Trial 175 in type 2 diabetes showed TI was superior to placebo for A1C reduction when used as an add-on to maximally tolerated doses of oral agents in insulin-naive subjects. The older trial 102, using Medtone plus basal, showed TI was non-inferior to twice daily premix. There was durable control over 52 weeks. In both type 1 and type 2 subjects, TI demonstrated a weight advantage compared to active comparators. And finally, TI causes less hypoglycemia, both total and severe. There were clinically significant reductions of incidence and event rates of hypoglycemia when TI was compared to insulin comparators. And now I'd like to introduce Dr. Nick Amin, VP of Clinical Development, who will present the TI safety data.
Good morning. My name is Nikhil Amin, and I'm Vice President of Clinical Development at Mankind Corporation. It is my pleasure to review the clinical safety data of Technosphere insulin. I will start with the general safety information obtained from our clinical trials first, and then I'll go into details about some areas of special interest as outlined on the slide here. The clinical program involved approximately 6,500 patients and healthy volunteers in the safety population. Of this, approximately 1,000 individuals participated in clinical pharmacology studies, and approximately 5,500 patients with type 1 or type 2 diabetes participated in the controlled clinical trials. As shown in the table below, approximately 3,000 patients were exposed to Technosphere insulin, referred as TI throughout the presentation. 290 patients were exposed to Technosphere placebo inhalation powder, which does not contain insulin, referred as TP, and approximately 2,200 patients were treated with comparator antidiabetic agents, which includes subcutaneous insulin alone, oral agents alone, or a combination of both. Now, looking at the exposure duration for these controlled studies, for TI group, more than 700 patients, and for comparator group, more than 900 patients with type 1 or type 2 diabetes were exposed between 12 and 24 months. In addition, 229 patients with type 2 diabetes participated in an uncontrolled extension study and were followed for up to 4 years. As safety findings did not differ in any meaningful way between patients with type 1 and type 2 diabetes, and also between patients treated with using Gen 2 and Medtone inhalers, I will focus on the totality of the safety data for this diabetes group combined. However, for respiratory adverse events, I will also review safety findings by Gen 2 and Medtone inhaler for a side-by-side comparison. So let us first look at overview of the safety data. Incidence of all causality treatment-emergent adverse events was higher in TI than T placebo or the comparator groups. Incidence of serious adverse events were comparable between patients receiving TI and the comparator. The incidence of deaths was also comparable between groups. No deaths were attributed to TI or the comparator drug. Note that AEs leading to early discontinuation were more common in TI and T placebo groups than the comparator. The difference was primarily driven by adverse events related to the respiratory system. This is not surprising, as the comparator group did not receive inhaled powder. Examining the most frequently occurring adverse events, the most common adverse event reported with TI was cough. Besides cough, oropharyngeal pain and throat irritation, not surprisingly, were more common with inhalation of a dry powder in the TI and T placebo-treated patients. Other adverse events were generally similar in frequencies between the treatment groups. Now, taking more detailed look at cough, as you can see in the table on the top, there were no meaningful differences in the incidence of cough in TI and TP group between Gen 2 and Medtone inhalers. Regardless of the inhaler, for those 27% of patients who reported cough occurred predominantly in the first week of the treatment and declined with continuous use as the patient got acclimatized to inhaling the dry powder. Cough was typically mild, non-productive, often a single event or intermittent events occurred within 10 minutes of dry powder inhalation. Patients described cough as a throat-clearing cough or feeling a tickle in the back of the throat after inhaling the dry powder. Adverse events of cough were not considered serious by investigators. 2.8% of TI-treated patients discontinued due to cough. In those patients, cough resolved within 1 or 2 days after the patient discontinued TI. No association was noted between cough and changes in lung function or the TI dose. Now turning to the serious adverse events, SAEs were comparable between patients receiving TI and the comparator. The most common SAE were reported in metabolism and nutrition system organ class, or SOC. Although not included in the slide, at the preferred term level, hypoglycemia was the most common SAE, as would be expected in insulin studies. The incidence of respiratory SAEs were low in both groups. No patient reported more than one respiratory serious adverse event. SAE in other organ system class groups were balanced between the treatment groups. For cardiovascular AEs, 2 post-hoc analyses called the broad and custom were conducted based on the investigator-reported AEs, and they are described in the sponsor's briefing document. Results of this analysis show no excess cardiovascular risk associated with TI use. Now, when looking at the discontinuation, we see that more patients in the TI group discontinue due to adverse events than the comparator. Not surprisingly, the difference was again driven primarily by respiratory events. When we examine the AEs leading to discontinuation at the preferred term level, the top 3 AEs leading to discontinuation were cough, dyspnea, and hyperglycemia. AE of dyspnea, or shortness of breath, was generally mild or moderate in severity and resolved quickly without any specific treatment. In 2 cases, dyspnea was considered serious. This occurred in a patient with a significant other condition, one in a patient with urinary tract infection, renal failure, and fluid overload, and in a second patient who has a hypertrophic cardiomyopathy and heart failure who reported orthopnea. Now let's move on to the examination of these specific topics, starting with pulmonary safety. Given the inhaled delivery system, pulmonary safety was evaluated with particular care, including side-by-side comparison of pulmonary safety between Gen 2 and Med 2 inhalers. So let us first look at respiratory adverse events by inhalers. As shown by the highlighted 2 columns on the left of the table, for the TI group, no meaningful differences were noted in the incidence of respiratory AEs between the 2 inhalers used to administer TI. Same is true for the inhaled TP group. Overall, respiratory AEs were more common in the inhaled treatment groups than the comparator. In addition to adverse events, the pulmonary safety assessment included imaging studies, chest X-ray at the beginning and end of the trial of 1 year duration or less, and annually in longer trials. A subset of 474 patients had serial HRCTs with blinded independent central review of all images. As imaging studies did not show any safety concerns, this presentation will focus on lung function. To minimize the variability in the PFT measurements, MannKind instituted a rigorous and standardized lung function testing and quality control program. Over 34,000 lung function tests were done during the clinical studies. Lung function tests included spirometry, body plethysmography, and lung diffusion capacity, or DLCO. All tests were done at the trained and certified PFT labs only and were conducted in accordance with the standards set by American Thoracic Society and the European Respiratory Society, and all tests were reviewed centrally by blinded independent reviewers. For today's discussion, in the interest of time, I'll focus on forced expiratory volume in 1 second, or FEV1, which is the most reproducible of all lung function tests. However, the results of the other lung function tests also yielded similar results. When discussing pulmonary function, it is important to note that we see a natural decline in lung function over time as a simple function of aging. To better understand this, let us start with our 2-year pulmonary safety study. In this study, patients were randomized to receive either TI or they continue their usual antidiabetic regimen without any inhaled drug. Study also included a cohort of non-diabetic subjects who did not receive any drugs to evaluate the PFT changes over time. For healthy non-diabetic individuals, the FEV1 should be approximately 3.5 liters. As would be expected, there was a small decline in mean FEV1 from baseline in non-diabetics group who did not receive any drug over a 2-year period, as represented here by the orange line. In the same study, the diabetic patients treated with usual care showed a slightly larger decline here in gray, which is a recognized feature of diabetes. Diabetic patients treated with TI in blue had a slightly larger decline at first visit, but importantly, thereafter the treatment group difference remained non-progressive or did not worsen for up to 2 years. The treatment group difference at month 24 was 37 milliliter. In this study, more TI patients dropped out early than the comparators. So examining the PFT changes in the completed population show the magnitude of FEV1 changes over 2-year treatment period was similar to the ITT population, and the treatment group difference at month 24 was 38 milliliter. The pooled data from phase 3 studies for both type 1 and type 2 combined patients showed similar magnitude and pattern of changes from baseline in FEV1 as did 030 study. At month 3, the decline in TI group was approximately 40 milliliter larger than the comparator. Thereafter, the treatment group difference remained relatively constant between 40 and 45 milliliter over 2 years. Several sensitivity analyses conducted to account for the dropouts supported the conclusions of the primary MMRM analysis. So what happens to the difference in FEV1 after TI therapy is stopped? To evaluate that, we obtained pulmonary function tests in about 600 patients who had completed a parent trial, one of the phase 3 studies of 6 months or 20 to 24 months duration. In these studies, there was a small treatment group difference in FEV1 at the end of the randomized treatment period, which is marked as a vertical red line in the slide. The small difference in FEV1 between the groups resolved by 1 month post-therapy. Examining the mean values alone may not reflect the distribution of changes in FEV1. So this curve on this slide shows the frequency distribution of patients with percent change from baseline to last measurements in FEV1, blue for TI and gray for the comparator. We can see that the mean changes in FEV1 values were not driven by outliers experiencing large changes, but rather by a slight shift to the left in the distribution of entire group with small changes. So in summary, in addition to the natural and expected decline in pulmonary function due to aging, there was a small additional decline in the TI group. This was observed early after treatment initiation, and thereafter the treatment group difference remained non-progressive for up to 2 years. Any difference between the treatment group disappeared rapidly after TI was discontinued. And though not presented here, we found no association between changes in lung function and age, gender, average daily dose of TI, and between patients who did or did not report cough during the trials. Now let us look at how the PFT changes compare between the Gen 2 and Medtone inhalers. Study 171 included both Gen 2 and Medtone treatment arms for a head-to-head comparison between the two inhalers as recommended by FDA. Changes from baseline shown in this figure in mean FEV1 over a 24-week treatment period is shown here. Blue line represents the Gen 2 inhaler arm, and the green line represents the Medtone arm. There was no significant difference in the magnitude and pattern of FEV1 changes between the two inhaler treatment arms. The small treatment group differences in FEV1 between TI and the SPAR groups at the end of the randomized treatment period again resolves by 1 month. Besides within-study comparison, how the FEV1 changes associated with TI compares with between this study and the previous Medtone studies. The side-by-side comparison of FEV1 changes by treatment groups in study 171, which is shown on the left in this table, and the pooled previous Medtone studies on the right are shown here. Top rows of the table summarizes baseline FEV1. Changes from baseline to week 12 in the middle row and week 24 in the bottom row by the treatment arm in study 171 are compared to changes observed at the same time points in the pooled previous Medtone studies. Comparator arm, who did not receive the inhaled drug, also showed similar magnitude of FEV1 changes from baseline to week 24, demonstrating the assay sensitivity and consistency of results. In summary, as reviewed by Dr. Baughman earlier, same TI powder formulation is used for both Gen 2 and Medtone inhaler. The fine particle dose, or the dose reaching the lungs upon inhalation, is also similar between the two inhalers. The results of the PK study showed equivalent systemic insulin exposure and FTKP concentration regardless of the inhaler used to administer the TI. In clinical studies, as I reviewed, no meaningful differences were noted in the safety profile, including respiratory adverse events and changes in lung function between the two inhalers. So when combined these findings, it clearly establishes that overall pulmonary safety profile in Gen 2 studies is comparable to the one noted in the previous Medtone inhalers. Now, the clinical efficacy and safety studies excluded patients with FEV1 of less than 70% predicted, current smokers, or those who quit smoking within 6 months of enrollment, and patients with asthma, COPD, or any other chronic lung disease. Given these exclusions, it is important to understand the safety of TI in patients with asthma and COPD. The short-term safety of TI in patients with asthma and COPD was first evaluated in 3 clinical pharmacology studies, 2 studies in patients with asthma and 1 in patients with COPD using the Medtone inhaler. Long-term clinical study of TI in patients with asthma and COPD using the Gen 2 inhaler is ongoing. So let us first look at the safety in asthma patients. When asthma patients receive a single dose of TI in study 113, 5 of the 17 patients developed adverse events of wheezing and asthma symptoms. 2 were serious, with FEV1 declining by 33% and 45% after TI administration. Both patients required rescue bronchodilator, but recovered with no further complication, but were discontinued from the study. In addition, the serial spirometry testing before and after TI dosing showed acute decline in mean FEV1 of approximately 12% or 400 milliliters, which recovered to baseline values by 120 minutes. When asthma patients, excluding those 2 patients who discontinued, received albuterol, a short-acting bronchodilator, 5 minutes before TI dose, no decline in FEV1 was noted post-dosing. From this study, we learned that TI is poorly tolerated by patients with asthma. So what about patients in COPD? In a small study of 20 patients with COPD, serial spirometry was performed in a subset of 10 patients to evaluate acute changes in FEV1 immediately post-TI inhalation. COPD patients experienced a mean decline of approximately 9% or 220 milliliters in FEV1 by 18 minutes after dosing and improved gradually over subsequent 8 hours. Based on this data, we concluded that TI is also poorly tolerated by COPD patients. Although the experience with TI in patients with asthma and COPD is limited, based on the available safety information and as mentioned in the FDA's briefing document, we propose contraindication in patients with asthma and COPD. Additionally, all patients should be carefully screened by medical history, physical examination, and spirometry to identify any potential underlying lung disease, including asthma and COPD, prior to initiating TI therapy. Note that this labeling and screening requirement will focus of our proposed REMS program to educate physicians about the potential risk associated with TI and importance of appropriate patient selection for the safe use of the product. Now let us move to the malignancies. First overall, and then lung malignancies. As you can see here, malignancies were observed in all treatment groups and show heterogeneity of tumor types with no predominance of a specific type of cancer or clustering by organ system. The anatomic location and type of cancers observed in the clinical studies were consistent with the types most commonly observed in the general population. Observed incidence of overall malignancies does not exceed the rate expected in general U.S. population based on the SEER database. As shown, there was one lung cancer case in TI-treated patient, a small cell lung cancer of neuroendocrine variety, and none in the comparator group. Now let us look at this case on the next slide. In the control study 102, a 62-year male with type 2 diabetes in the TI group was diagnosed with a small cell lung cancer 137 days after randomization. An additional case of lung cancer was diagnosed in an uncontrolled extension trial. A 68-year-old male with type 2 diabetes was diagnosed with a non-small cell bronchogenic carcinoma. In this case, patient had a family history where father also died of similar lung cancer. In both these cases, patient had a history of heavy smoking. Taking both these lung cancer cases into account, one in the control study and one in an uncontrolled extension study, the observed rate of lung cancer associated with TI is 0.8 cases per 1,000 patient year of exposure. This rate is consistent with the 1 to 2 cases per 1,000 patient years seen in diabetic population made up of both smokers and non-smokers. In addition, there were 2 cases of lung cancers reported after completion of the clinical studies. We received 2 voluntary reports of lung cancer diagnosed approximately 2.5 years and 3.5 years after having been treated with TI in clinical studies. In the absence of a systematic follow-up and information related to exposure to any new risk factors since trial completion, it is difficult to ascertain causality or estimate the incidence rate as both numerator and denominators are uncertain. So both preclinical data, as discussed by Dr. Baughman earlier, and clinical data do not indicate increased risk associated with TI for either lung cancer or overall malignancies. While we find this reassuring, the total number of reported cases are too few to definitely assess the long-term risk. So MannKind is committed to conducting a post-approval long-term prospective study to evaluate any potential risk of lung cancer associated with chronic use of TI. Now let us look at DKA, or diabetic ketoacidosis, other safety event of interest where numerical imbalance was noted between TI and comparator group in the original submission. As shown here, there were more cases of DKA in the TI group, all reported in the original submission and all considered not related to the study drug by the investigators. When evaluating individual cases, we found that in situations with intercurrent infections or illnesses, such as gastroenteritis, flu-like illnesses, urinary tract infection and gluteal abscess, cholecystitis, hepatitis, pancreatitis, or worsening of the patient's underlying depression, this patient either stopped taking or skipped their insulin doses. DKA cases occurred as early as 3 days after randomization to more than 400 days after treatment initiation with no apparent temporal pattern. We found no association between DKA and anti-insulin antibody response. The 3 TI-treated patients were discontinued because of DKA. As a result, we emphasize in our subsequent clinical studies that under such circumstances as intercurrent illnesses, insulin treatment must be continued. Frequent monitoring of blood glucose and dose adjustment may be required like any other insulins. If administration by inhalation is not feasible, substitution with an injectable insulin may be necessary. Following this enhanced site education, no new cases of diabetic ketoacidosis have been noted in the clinical studies since 2009 original submission. Now I will turn to our plans for risk management activities after approval. In order to continuously ensure and evaluate the safety of TI, we propose the following. The label is the basis of risk management plan and will drive appropriate patient selection by contraindicating in patients with diagnosed chronic lung disease and screening for undiagnosed lung disease by detailed medical history, physical examination, and spirometry prior to usage. It will also guide to carefully reevaluate patients if they experience any respiratory events such as persistent cough, wheezing, or breathing difficulties. The REMS program will reinforce the labeling and appropriate patient selection with the focus on informing healthcare providers of potential pulmonary risk, including changes in PFT over time, concerns about use in inappropriate patient population, and potential for breathing difficulties in patients with chronic lung disease. A long-term prospective study is proposed to evaluate the potential risk of lung cancer associated with TIUs. This study will also evaluate risk of other malignancies and respiratory, allergic, and serious hypoglycemia events or other events with low incidence or long latency. The enhanced pharmacovigilance plan is also proposed to monitor targeted medical events such as DKA, eye events, and respiratory events for signal detection and evaluation. So in conclusions, the safety of TI has been extensively evaluated and well characterized in clinical studies. The most common adverse event, excluding hypoglycemia, is dry, transient cough. The observed small reduction in lung function, regardless of the inhaler, are noted early within first 3 months, but thereafter remain non-progressive. It is clinically reassuring that the PFT changes are non-permanent and the treatment group differences resolve upon cessation of TI. Overall safety profile, including the pulmonary safety, is comparable between the two inhalers. The observed lung cancer rate is consistent with the expected rate in diabetics population. Overall, Technosphere insulin is well tolerated in the treatment of hyperglycemia in patients with type 1 or type 2 diabetes. Now I would like to invite Professor Garrick to provide his clinical perspective of Technosphere insulin. Dr. Garrick.
Thank you. Good morning. I'm Dr. John Garrick, Professor of Medicine Emeritus at the University of Rochester School of Medicine, and I'm pleased to be here this morning to provide my clinical perspective on the benefit-risk profile of Afrezza. Afrezza is a new system for insulin delivery. The inhaler is new, but the drug is regular human insulin, which we've been using for about 30 years. Afrezza behaves differently than other prandial insulins because its PK profile is different. I believe that the data presented this morning indicate that Afrezza has a favorable benefit-risk profile. A unique feature of Afrezza is its PK profile. It has a fast onset of action, closer to the physiologic insulin response than any other exogenous insulin. I believe this has important clinical benefits. The rapid onset and offset is better synchronized with mealtime plasma glucose excursions. This results in less hypoglycemia in the 2 to 5 hour period following meal ingestion. Furthermore, it would be expected that any hypoglycemic episodes that would occur in patients taking Afrezza would be of lesser severity due to its shorter duration of action. Afrezza also provides dosing flexibility. It can be taken up to 20 minutes after the start of a meal, and if needed, a supplemental dose can be taken between meals. You've seen the efficacy data. The primary endpoint was met in both type 1 and type 2 diabetes trials. In trials of people with type 1 diabetes, Afrezza's treatment effect was not as large as Aspart's, but not unacceptably worse either. And comparable results were obtained in other trials of prandial insulins in people with type 1 diabetes. In the studies of people with type 2 diabetes, there was a significant decline in mean HbA1c level. I anticipate that improved results will be obtained after approval, very similar to what we saw with the introduction of the rapidly acting analogs. Clinicians and patients will gain more experience with Afrezza and learn to optimize its titration. Afrezza addresses two important concerns for patients with diabetes who are treated with insulin, namely hypoglycemia and weight gain. The American Diabetes Association Working Group on Hypoglycemia has declared that even a 10 or 20% reduction in severe hypoglycemia or a 30% reduction in overall hypoglycemia represents a clinically important improvement. With Afrezza, there was a 43% reduction in event rates for severe hypoglycemia in type 1 diabetes, and a 65% reduction in type 2 diabetes, based on older active comparator studies. This reduction in hypoglycemia was still observed when achieved HbA1c levels were taken into consideration. In addition, the trends in weight with Afrezza were also favorable. Now, let me digress here for a moment. I have had considerable personal experience with hypoglycemia. Most of my research career has involved in causing hypoglycemia in normal volunteers and people with diabetes. And I was not overly impressed with moderate hypoglycemia until I myself came down with type 2 diabetes and developed hypoglycemia as a result of my treatment. Although my blood glucose went down only to about 50 and was rapidly reversed with a sandwich, it disrupted my whole day's work. And to tell you the truth, I stopped my diabetes medication because of it. So from personal experience, I know that even moderate hypoglycemia is very disrupting, and its avoidance is something that would be very welcome. Now, Afrezza addresses barriers to insulin initiation and intensification that Dr. McGill spoke about and can help overcome the clinical inertia associated with insulin therapy. As a non-injectable system, it provides another option for the 50% of people with type 1 diabetes who are not on pump therapy, who may want to, and for those who may want to have fewer injections. People with type 2 diabetes may find it easier to transition from oral therapy to inhaled insulin than to injected insulin. As we saw in the video that Dr. Boffman showed you, Afrezza can deliver a dose of insulin quickly, easily, and discreetly. It can be used in public without any fear of embarrassment. As we have seen, Afrezza has a manageable and predictable safety profile. The most common adverse effect is a mild cough that occurs after inhalation. It causes some discontinuations, but is only a minor annoyance in most patients and diminishes with constant use. And patients can be counseled on expecting this side effect. Afrezza is poorly tolerated in patients with diagnosed asthma, chronic obstructive pulmonary disease, and chronic lung disease, and I support the contraindication in these patients. I also agree with the requirement for spirometry to rule out any underlying lung disease. This seems reasonable and not particularly burdensome. There's been no lung cancer signal to date, either in the non-clinical studies or in the clinical program. To address long-term safety, the sponsor has proposed labeling and a REMS to guide patient selection, and a post-approval study to assess lung cancer and other pulmonary risks. I support both of these efforts. Afrezza can be used as a useful component in an individualized treatment regimen for patients with diabetes who need a prandial insulin. In people with type 1 diabetes, I would use it in patients especially prone to hypoglycemia and those on a prandial insulin who wish to lessen their injection burden. In people with type 2 diabetes, I would consider it for patients on a basal regimen who require intensification with the prandial insulin, as an entry insulin for patients failing oral therapy who are injection averse, and most importantly, in patients on one of the other prandial insulins who experience frequent hypoglycemia. In conclusion, then, the safety and efficacy data presented this morning demonstrate that Afrezza has an acceptable benefit-risk profile and offers an important treatment option for patients with diabetes. Thank you.
Mr. Chairman, that concludes our presentations. We are available to answer any questions the committee may have. And I'd also like to take at this time a moment to say that should a member of the committee want to inspect a device, we have some of our device engineers that are here with devices that perhaps could be done during a break or at lunch.
Okay, thank you very much to the sponsor for those very informative presentations. So we have a few minutes for discussion, or rather for clarifying questions. We'll have more time for discussion later in the day. And in terms of your questions, if you would remember to please state your name for the record before you speak. And then also, if you have questions, some people I see raising their hands already, if you signal Dr. Abraham Burrell here to my left will record those, and we'll try to get to people in order. If we miss you in this section, we'll try to get back to you later in the day. So we'll start with Dr. Calhoun.
Thank you, Bill Calhoun. I have two questions for clarification and then one comment, and we'll have a chance to take a look at the comment during our discussion time. The first question is on the design of study 171. As I understand, that was not a blinded study, so you didn't use a dummy inhaler and you didn't use a dummy injection.
That's correct,
Correct.
sir.
And so it's at least conceivable that people who were on injection versus on inhaled insulin might have subtly altered their dietary intakes, and you'd have a confounding factor that's unmeasured. And I didn't hear you talk about the possibility that you might have a confounder there.
Yes. And to address that, I'd ask Dr. Bregman to return to the podium and address your comment.
So as Dr. Boffman said, this was an open-label study, as you recognized. It would be unethical to have a double dummy design, we felt, because people are constantly injecting, and to do that, both arms would have to take inhaled as well as injected. And I think it's standard to have open label in an insulin an insulin comparator study of this sort. As you indicate, theoretically, the potential for confounding does exist. However, the primary endpoint is an objectively measured A1C. And furthermore, dosing was observed by an independent titration monitoring committee that was independent of the company and ensured that the delineated titration algorithms in the trial were followed at the site level.
And so the second quick technical question is, acid is a known instigator of cough, particularly in those who've got airway responsiveness, asthma, and COPD. And if I recall correctly, your manufacturing included precipitation of the FDKP at pH 6. So do you have any pH modeling of the material when it gets into airway lining fluid? Do you know what happens to airway pH?
We have not specifically studied airway pH following inhalation. Again, the powder goes into solution when it reaches 6.5. But the actual pH of the interstitial fluid or the lung fluid has not been measured in our trials.
And then the last comment is simply that a normal spirometry would probably be inadequate to exclude the diagnosis of asthma, which is can be a tricky diagnosis to establish or particularly to rule out. And we can talk about that more.
Dr. Cook?
So for both of the recent studies, there was this allowance for rescue dosing if the 90-minute postprandial glucose level was high in the TI groups. What were the average number of insulin doses per day in those inhaled patients? The injection patients presumably are getting the 3 doses of prandial insulin plus their basal. But what's the average number of doses per day to get a feeling for sort of how much rescue was required or utilized to achieve the A1C that they got?
Yes. Yes, I'll ask Dr. Bregman from clinical again to address your question.
So in trial 171, as you indicated, the subjects in the aspart arm had basal plus aspart. The subjects in the TI arm had basal plus inhaled. The average number of doses was of regular doses was 1 or 2 cartridges per meal per patient. About 75% of the subjects used 1 or 2 cartridges per meal per patient. Now, with regard to what you called rescue, I'm pretty sure what you're referring to are the supplemental doses that could be administered.
Right.
So these could be administered if the if the self-monitored blood glucose level at 90 minutes was 180 mg per deciliter or greater. A one-time supplemental dose could be administered, and we have data for that. Slide up. It turns out that the e-diary data using the SMBG inputs showed that such values occurred in the TI Gen 2 and then the safety Medtone arm about 40% of the time. However, out of those 40%, when a supplemental dose was expected, it only occurred about 20% of the time. We were a little surprised ourselves that there wasn't more use of the supplemental dose, and we suspect that it was because of unfamiliarity with this concept. But there were some who used it. They did not experience more hypoglycemia. However, the results of our trial would probably need to be based on those who were using the mealtime doses, because there wasn't that much use of the supplemental doses. We expect that as patients become more familiar, they might use it more.
And then just one other simple question. A number of comments have been made about the ability to take these doses up to 20 minutes after meal initiation. But is there any data to support that in terms of clinical efficacy?
A study was conducted, a pharmacokinetic study was conducted to evaluate glucose response post-meal when dosed 15 minutes prior to, at the time of the first bite of food, and 10 and 20 minutes subsequently. And that data supported the label in the trials. In the 171 study, we were able to analyze the dosing time distribution relative to the meals. And I'll again ask Dr. Bregman to present that data.
We did gather that data, and I'll show you a histogram which describes it. Slide up, please. Basically, about 80 to 90% of the dosing actually took place within 5 minutes before or after the meal. There was about 10 or 20% that was used greater than 10%, greater than 10% after 10 minutes after the meal, excuse me, or less than 10 minutes before the meal. So most of our data shows that patients were taking it around meals. Again, I suspect it's because that's what patients are familiar with, and we didn't have enough, people at extremes to look at whether or not that affected the efficacy.
Dr. Samplin.
So I have two questions relating to dose and dosing. The question with respect to dose is, as I understand it, in your trials, you assumed a 4-unit sub-Q insulin equivalence to a 10-unit cartridge. And I'm sorry, yeah,
You would say.
a 4-unit conversion, but you're recommending, I gather, in the product labeling, it's going to be a 3-unit. So where did that change occur? And what was the basis for that?
Yes, that conversion for your first dose of Technosphere insulin was initially based on for every up to 4 units of aspart would be converted to a 10-unit cartridge. Subsequent to that time, we conducted the 171 and 175 studies, as well as the 176 clinical pharmacokinetics study. If I could have this slide up, please. What we see here on the left was our assessment of that data subsequent to those trials. On the left, you see the pharmacokinetic data reporting relative bioavailability from the 176 and 177 studies, data that's included in your briefing book. And we see we have a relative bioavailability of about 30%. So as such, a 10-unit cartridge would be comparable to 3 units of subcutaneous insulin. The 20-unit cartridge would be comparable to the 6-unit. When we evaluated the dosing from the phase 3 study in type 1s, the 171 trial, we saw that the average dose of Afrezza was 102.7 units. The average dose for insulin aspart was 25.5 units. That ratio is 25%, so approximating 3 units of subcutaneous insulin per 10 units of insulin loaded into the cartridge. Subsequent to that, we evaluated the pharmacodynamic response from the 176 study. And when we evaluated that model and then converted the regular human insulin effect at the same doses used for Technosphere insulin, we saw that consistently we had about a 33% of the response. So we now have 3 classic methods for characterizing relationship between different routes of administration or different dosage forms, and we came to the conclusion that the patients are best served by having a dose transfer of 3 units for a 10-unit cartridge.
And just a second part of that question, you mentioned that the average number of cartridges used for any given prandial treatment was 1 or 2. What would be the maximum number of cartridges someone might have to use, and how long it might have taken to fumble through getting those into the device and inhaling them? And would that affect the PK, for example?
Yes, sir. We have dosed multiple cartridges. We have dosed multiple cartridges. We went up to 80 units, which would be 4 cartridges in our pharmacokinetic trial, and we saw little change in the time of the peak concentration, some broadening, but only by a couple of minutes. The average dose in the 171 trial was 102 units of Technosphere insulin. That's per day, so that would average about 33 units per meal, which is easily accomplished with two cartridges.
This is Robert Smith. I have a question that is somewhat related to dose, and it specifically relates to the occurrence of diabetic ketoacidosis. When I looked over the data, perhaps you'll do a better job than me, I found 12 cases of DKA where I was able to look at the clinical details. 3 of those were 2 patients who very early developed DK. There were questions about their proper use of the device. One had a case of acute cholecystitis. So I want to exclude those from the question I'm going to ask you, because the other 9 had a mean insulin dose, daily dose of 200 by my calculation, 200 units per day. So that would be twice the mean that you've been reporting. I assume they were, for the total group, I assume they were all type 1 diabetics. So my question, and you may want to look over some numbers and come back later today to talk about this, perhaps this afternoon, but my question is whether high dose, for example, in this range of a mean of 200, in fact defines a high-risk group for DKA. and this perhaps linked to the issue of maximizing efficacy, which we've also seen perhaps occurs with a dose of 60 or above. So if you have a quick answer, okay, or actually I think what I'll ask is if you will come back, because I'd like to get another question in, and we can perhaps address this question, but if you have some work to do on that to pull data together, I'd be interested to see that.
Yes, sir, we'll do that. Thank you.
Dr. Britton.
Yeah, my question is about both slides CE 39 and CE 40. I'm not sure which one to look at first, but these are very interesting and very helpful. On the other hand, particularly CE 40, where you're adjusting for a post-baseline characteristic, it's a little hard to interpret. And CE 39, even though it's completely legitimate to do what you did, it's still a little hard to interpret because you're sort of stratifying on a post-baseline characteristic. So, you know, if I were a patient, I would want to know something a little bit different, which is, you know, what proportion of the time am I in the targeted range? Or define like an optimal outcome being, you know, having a good hemoglobin A1C and having a low hypoglycemia rate. Did you do any analyses that look at it from those kinds of angles?
Let me ask Dr. McGill to come to the podium and address that. She was involved with both of our trials and would be able to comment certainly on the patient perspective.
So I'm not going to answer your question about the analyses, but we can perhaps delay that for later to balance that time spent in range versus time spent out of range. I do have a comment, though, about this difference, particularly in the type 1 trial, that the difference in A1C, the 0.19%, equates to, in blood glucose terms, 7 milligrams per deciliter. 7 milligrams per deciliter is an imperceptible amount both to patients, short term, we don't adjust insulin usually over that area, and long term with respect to complications. It's a very, very small delta. However, the difference in hypoglycemia was immediately felt by patients, was appreciated by the patients who, and they told us, we know we're having less hypoglycemia. So exactly how it lays out, time in range and time out of range, I can't tell you the answer, but I can tell you that difference of the comparison of that small difference in A1C versus that rather large difference in hypoglycemia was really highly clinically relevant to our patients.
So for time reasons, we're going to need to take a break now and move on. I know there are several of you on the panel who haven't had your opportunity to address your questions, and I'll come back to you later today when we have our next question session. So we're going to take a 10-minute break now. Panel members, please remember there should be no discussion of the meeting topic during the break amongst yourselves or with any member of the audience. And we're going to resume at 10:28 sharp. So please be back in your seats by then. Okay, I would like us to get started, so we'll now proceed with the FDA presentations.
Good morning. My name is Lokesh Jain. I am team leader for the diabetes team in the Office of Clinical Pharmacology. The primary reviewer for this application is Dr. Sang Chung, who is also present here. In this presentation, I will provide the clinical pharmacology background to facilitate the discussion on following issues. First, the lower efficacy of Afrezza compared to that observed for subcutaneous mealtime insulins. Second, the adequacy of evidence for the proposed dosing regimen for switching from subcutaneous to Afrezza insulin. And finally, whether dose titration for Afrezza provides incremental benefit at higher doses. My objectives are to present pharmacokinetic and pharmacodynamic characteristics, hereafter referred as PK and PD, of Afrezza relative to injected mealtime insulins. Dose proportionality and between subject variability for PK and PD parameters. A discussion on the proposed dosing regimen, followed by summary. I will primarily focus on data from two single-dose studies, 176 and 177. These studies were submitted in the recent complete response resubmission and were conducted with the to-be-marketed Gen 2 device. 176 is a clamp study in healthy subjects in which PK and PD were characterized for 4 doses of Afrezza, 10, 30, 60, and 80 units, which are approximately equal to 4, 12, 24, and 32 units of regular human insulin. These were compared against a single 15-unit dose of subcutaneously administered regular human insulin. 177 is also a clamp study, but in subjects with type 1 diabetes. In this study, PK and PD for 20 units of Afrezza, which is approximately equal to 8 units of Lispro, were compared against 8-unit dose of subcutaneously administered insulin Lispro. Before I go into the PK-PD profile of Afrezza, let me review the insulin secretory patterns in normal healthy individuals to help put the PK-PD profiles of Afrezza into perspective. The figures on this slide are taken from a publication by Polonsky et al. For simplicity, I would like to draw your attention to only the profiles in solid lines, which represent the physiological release profiles in normal healthy individuals. The figure on the left shows the mean profiles of glucose measured over 24 hours following administration of 3 mixed meals. Glucose is shown on the y-axis and time on the x-axis. This figure shows that glucose absorption after each meal lasts for about 4 hours. The figure on the right displays the corresponding plasma insulin concentration profiles over 24 hours and shows that post-meal insulin secretion mimics the glucose absorption profile and also lasts for about 4 hours. This balance between glucose absorption and insulin release and the adequate insulin coverage for the entire post-meal glucose absorption period may be important to ensure adequate glycemic control. Now I will discuss the PK-PD characteristics of Afrezza. This slide shows the plasma insulin concentration time profiles for Afrezza delivered by the Gen 2 device in healthy subjects from study 176. Going from bottom to top, shown here are profiles for 10 units Afrezza in green, 30 units in purple, 60 units in blue, and 80 units in orange. Again, which are equivalent to regular human insulin doses of approximately 4, 12, 24, and 32 units. Note that peak insulin concentrations increase in an approximately dose proportional manner from 10 to 80 units dose. But in contrast to the natural release profile in normal individuals, insulin concentrations decline back to about baseline levels within 1.5 to 3 hours. This duration is shorter than the duration of post-meal glucose absorption for normal healthy individuals shown in the previous slide. These profiles are compared against the PK profile for 15 units dose of regular human insulin shown in red color. Note that insulin concentrations for subcutaneously administered insulins, regular human insulin, return to baseline in about 6 hours, providing sufficient coverage for the entire post-meal duration. It is clear that insulin concentrations for Afrezza dose of 30 units and above are considerably higher than subcutaneous insulin. Similar differences in PK were also observed in type 1 diabetic subjects. In blue is the profile for Afrezza 20 units, and in red for insulin Lispro 8 units. Overall, PK profiles for subcutaneously administered insulins appear to be more close to the physiological release profile compared to Afrezza. The observed differences in PK also translated into PD differences. The figure shown here is from study 176 in healthy subjects. In this CLAMP study, PD effect was measured as glucose infusion rate, abbreviated as GIR, which is a measure of how much glucose is removed by insulin. The red profile is for subcutaneously administered insulin, and the other 4 profiles are for increasing doses of Afrezza. First, note that the time to maximum PD effect is earlier, and the duration of PD effect is shorter for all Afrezza doses compared to subcutaneous insulin. Second, if you recall from the previous slide, insulin concentrations for the 30-unit dose of Afrezza were considerably higher than subcutaneous insulin. But it does not translate into higher GIR effect. GIR profile for 30-unit Afrezza is shown in green color and subcutaneous insulin in red. The figure on the right is from study 177 in type 1 diabetics, drawn to the same scale as left figure. You are seeing in blue GIR profile for 20 units Afrezza and in red for 8 units Lispro. Subjects with type 1 diabetes have minimum interference from endogenous insulin, and the observed PD effect in this population is a better reflection of the effect of exogenously administered insulin. Again, recall from the previous slide, the insulin concentrations for Afrezza were significantly higher than insulin Lispro. However, from this figure, it is evident that it did not translate into higher glucose clearance in type 1 diabetics. The maximum effect of Afrezza was achieved by about 50 minutes and then declined to low baseline levels by about 160 minutes. In contrast, the effect of Lispro was maintained for about 4 to 6 hours. For comparison, this slide shows the glucose infusion rate versus time profiles for prior approved inhaled insulin Exubra relative to subcutaneously administered regular human insulin and insulin Lispro. Note the duration of GIR effect. Unlike Afrezza, the duration of GIR effect for Exubra is comparable to subcutaneously administered insulins. Next, I will discuss the dose proportionality for PK and PD based on results from study 176. PK PK exposures increased in a dose proportional manner, meaning with an increase in dose, there was a corresponding increase in PK exposure. These data are presented in detail in the FDA backgrounder. However, on the PD side, with increase in dose, a less incremental benefit in PD response was seen. Remember this figure from previous slides? The PD data from this figure were plotted against the PK exposures to have a better understanding of the PK-PD relationship. We have here PK on x-axis, that is insulin AUC for 0 to 180 minutes, and PD on y-axis, that is GIR AUC for 0 to 240 minutes. Open circles represent the 10-unit dose of Afrezza, plus symbols represent 30 units, X symbol represent 60 units, and triangles represent 80 units. As you note from this figure, with increase in insulin concentrations, there is an increase in GIR response. However, after the 60-unit dose, there appears to be a less incremental dose-related benefit, evident from the best fit line in this graph. The less incremental benefit with increase in dose is also known for subcutaneously administered insulins. However, since only one subcutaneous dose was used as a comparator, it is unknown if the diminishing benefit at higher doses of Afrezza parallels to that seen with subcutaneous insulins. One may wonder if the point of diminishing PD effect may have reached early for Afrezza compared to the subcutaneous insulins, and is important to consider while reviewing results from phase 3 trials. This is further emphasized by the results of clinical study 005 submitted with the original NDA and conducted with the Metton device. 4 doses of Afrezza, 14, 28, 42 and 56 units were tested against placebo in type 2 diabetes for a 12-week duration. Change from baseline in HbA1c and placebo-corrected change in HbA1c are shown in third and fourth column respectively. The placebo-adjusted change in HbA1c was minus 0.5% with the 14-unit dose, but remained between 0.7 to 0.8% for 28 to 56 units doses. Therefore, this trial was not able to demonstrate dose-related incremental benefit in HbA1c response. Next, I will discuss the between-subject variability. These are the same PK/PD data from study 176. The between-subject variability for Afrezza PK increased in a dose-dependent manner. Coefficient of variations for PK exposures are shown in this figure. First 4 bars are for Afrezza doses from 10 to 80 units, and the last bar represents variability for 15 units of regular human insulin. Large PK variability at higher doses of Afrezza may be because of the need for administering multiple cartridges and differences in inhalation flow rates between actuations. Conversely, the between-subject variability for PD decreased with increase in dose, shown in red color bars. It is possible that this finding is because of diminished incremental benefit in PD response at higher doses. Last topic of discussion is dosing regimen. Shown here is the applicant's proposed dosing regimen in the prescribing information for switching from prandial subcutaneous insulin to Afrezza treatment. It assumes an approximate ratio of 3.4 to 1 between the doses for Afrezza and subcutaneous insulin. With this dosing regimen, a patient on a 3-unit dose of subcutaneous insulin will receive an approximately 10-unit dose for Afrezza. Note that this dosing regimen is different than the dosing regimen applicant tested in phase 3 trials, shown in the rightmost column. Ratio of Afrezza to subcutaneous insulin dose was approximately 2.5 to 1 for phase 3 trials. If you compare these two dosing regimens, you will notice that a patient on 16 units of subcutaneous insulin was switched to 40 units of Afrezza in phase 3 trials, and the same patient will receive 60 units of Afrezza with the proposed dosing regimen. There are some common concerns regardless of which dosing regimen we look at. A similar dose-response relationship is assumed between Afrezza and subcutaneous insulin, such that the same dosing conversion ratio is applied across the entire dose range. Applicant only tested one subcutaneous dose as a comparator, so there is not sufficient information to support this assumption. Also, it is unknown if the dose at which we start seeing diminishing PD response for Afrezza parallels that for subcutaneous insulin. This may have implications on dosing titration. The applicant justified the proposed dosing regimen based on the mean PK bioavailability for Afrezza of about 24 to 33% relative to subcutaneous insulin. Other justification was comparison of mean daily prandial doses for Afrezza versus subcutaneous insulin from phase 3 trial in type 1 diabetes. There are limitations to this comparison, such as differences in basal insulin dose and titration schemes between the two arms, and generalizability of insulin aspart data to regular human insulin and other analogs. Finally, I will summarize the findings. PK and PD profiles for Afrezza are different from subcutaneous insulin. Maximum PD effect for Afrezza occurs early, but the duration of PD effect is shorter compared to subcutaneous insulin. A less incremental benefit is observed at higher doses of Afrezza. It is unknown if the diminishing PD benefit seen at higher doses of Afrezza parallels to the dose response for subcutaneous insulin. Finally, the dosing regimen proposed by the applicant makes some assumptions which are not well supported, such as the assumption of similar dose response between Afrezza and subcutaneous insulin. This concludes my presentation. Now I will turn the podium to Dr. Lisa Yanoff.
Good morning. I'm Lisa Yanoff, clinical reviewer in the Division of Metabolism and Endocrinology Products. The focus of my first presentation today is the clinical efficacy of Afrezza. I will begin with a brief history of the Afrezza development program and overview of results from previously conducted major studies. I will then discuss the two new trials, 171 in type 1 diabetes and 175 in type 2 diabetes. And my talk will end with a brief summary and concluding remarks. The current review is the third time that FDA has considered the marketing application for Afrezza. In 2009, the applicant submitted the original new drug application seeking approval to market the Medtone device. The application included one major trial in patients with type 1 diabetes and two in patients with type 2 diabetes. The application was not approved during its first review for several reasons, one of which was that FDA did not believe that the efficacy of Afrezza was clearly established. The applicant's resubmission in 2010 sought approval to market the Gen 2 device. The application was not approved, in large part because there wasn't sufficient information about the new device. FDA informed the applicant that a future resubmission should include two new trials with the Gen 2 device, one in patients with type 1 diabetes and the other in patients with type 2. FDA requested that one of these trials include a treatment arm using the Medtone device so that we could directly compare pulmonary safety between the two devices. The focus of this presentation, then, is the efficacy data for the two new trials, 171 and 175. However, before moving to those trials, I will give a brief overview of results from the previous major studies. This slide shows the primary efficacy results of the three major trials submitted with the original NDA. Although these trials were conducted with the Medtone device, the old and new device have been bridged such that these trials can be considered supportive evidence of efficacy. In discussing efficacy results, I will use the abbreviation CI when referring to the 95% confidence interval of the estimated treatment difference between Afrezza and comparator. Starting on the left side of the figure, in the one-year trial 009 in type 1 patients, Afrezza was studied against aspart on a background of insulin glargine. Afrezza was found to be worse than aspart because the lower bound of the CI was greater than 0. Afrezza was also not non-inferior to aspart because the upper bound of the CI was 0.4%. Shown in the middle of the figure, in the 6-month trial 014 in type 2 patients, Afrezza was studied against aspart on a background of insulin glargine. Afrezza was worse than aspart because the lower bound of the CI was greater than 0. In the one-year trial 102, Afrezza plus glargine was non-inferior to the 70-30 insulin comparator. In considering these data, it is important to note that 70-30 may be a suboptimal regimen for some patients. 70-30, in contrast to Afrezza, covers only 2 meals of the day and does not allow patients to optimize the mealtime dose. A further concern in interpretation of the data from Trial 102 is that in the absence of a glargine-only arm, it is difficult to estimate the contribution of Afrezza to the overall glucose-lowering effect in the trial. The FDA's decision of complete response to the original NDA was due in large part to the efficacy findings of these studies. Now moving on to the new trials. We will start with Trial 171, conducted in type 1 patients. After giving a brief reminder of the trial design and primary results, I will mainly focus on the handling of missing data and insulin titration, which are important trial conduct issues that may affect the interpretation of the study results. Participants started the study with a 4-week basal insulin optimization phase, during which subjects titrated their basal insulin to reach protocol-specified goals. Subjects were randomized 1:1:1 to Afrezza with the Gen 2 device, Afrezza with the Medtone device, or insulin aspart to be used as their prandial insulin. The subjects then underwent a 24-week treatment phase consisting of 12 weeks of prandial insulin optimization and 12 weeks of stable insulin dosing. The primary study objective was to demonstrate that Afrezza, administered using the Gen 2 device plus basal insulin, is non-inferior with non-inferiority margin 0.4% to insulin aspart plus basal insulin in its effect on hemoglobin A1C. The Afrezza Gen 2 group was compared with the Afrezza Medtone group primarily to evaluate pulmonary safety. In the trial, patients stayed on their pre-enrollment basal insulin. Basal insulin dose adjustment was based on a subject-driven titration algorithm. The goal was to reach a fasting glucose between 100 and 120 milligrams per deciliter. And further details of the algorithms are provided in the backgrounder. The main focus of this slide is to emphasize that aggressive subject-driven titration algorithms for basal insulin were included in the protocol. This should, in theory, allow for subjects to approximate American Diabetes Association recommended glycemic targets. The plan for prandial insulin dosing and titration is shown on this slide. The key point here is that aggressive subject-driven titration algorithms for prandial insulin were also included in the protocol design, allowing for subjects to titrate prandial insulins to approximate ADA glycemic targets. This slide summarizes the subject characteristics of the study population. The key message is to emphasize that the population studied in Trial 171 was generally healthy without severe diabetes complications or recent severe hypoglycemic episodes. They were not primarily of advanced age and were not selected to be high risk for cardiovascular disease. Therefore, one may argue that investigators should have been comfortable following the titration algorithms to achieve A1C values of less than 7% or even less than 6.5% if desired. I will now outline the subject disposition for Study 171. 518 subjects were randomized to the trial. 174 were randomized to Afrezza Gen 2, 174 to Afrezza Medtone, and 170 to insulin aspart. As you can see, the dropout rate of 21 to 25% in the Afrezza groups was higher than the 11% observed in the aspart group. Note that the observed rate in the aspart group is more consistent with what is expected in such a trial. This somewhat large and differential dropout rate is important for missing data considerations in the primary efficacy analysis and will be discussed in subsequent slides. The discontinuations were more often for adverse events, primarily cough, physician decision, and withdrawal by subject. Looking further at the explanations for subjects who prematurely discontinued in the Afrezza arm due to physician decision and withdrawal by subject revealed 5 discontinuations for what appears to be inadequate efficacy and 1 additional adverse event. Of note, a higher rate of discontinuation among Afrezza-treated subjects, most often for adverse events and inadequate efficacy, is similar to what was observed in the clinical trials submitted in the original NDA. The primary efficacy analysis results for Study 171 are shown here. The baseline A1C for both groups was around 8%. The mean reduction in A1C from baseline to week 24 in the Afrezza group was 0.20%, which was less than the 0.42% reduction observed in the aspart group. The treatment difference was 0.22%, and the CI was 0.08% to 0.37%. The non-inferiority of Afrezza to aspart was demonstrated since the upper bound of the CI was less than 0.4%, the predefined non-inferiority margin. However, because the CI was entirely greater than 0, Afrezza was statistically worse than aspart on A1C change. This slide shows the change in A1C for completers on the left versus dropouts on the right. Recall that the dropout rates were 25% in the Afrezza Gen 2 group and 11% in the aspart group. Missing data add uncertainty to the results. This uncertainty is not addressed in standard analyses, which treat the missing data as ignorable, or as ignorable after accounting for covariates. The concerns intensify the greater the amount of missing data. The FDA statistician's analysis using the completers cohort had similar findings to the primary analysis based on the overall population. However, the dropouts in the Afrezza arm had mean increases in A1C during the 12-week titration period, while mean decreases were observed in the aspart arm. Based on these data, the missing at random assumption used in the applicant's primary analysis does not hold for the Afrezza arm. One may question whether the overall treatment difference would have been larger than the 0.22% difference shown in the primary analysis if all the dropouts had stayed in the study. FDA requested that the sponsor perform a sensitivity analysis that would help further evaluate the impact of missing data. Analysis 1 in this table is the analysis requested by FDA. This method involves multiple imputation under the non-inferiority null, which includes adding 0.4% to the imputed week 24 A1C value for all discontinued subjects in the Afrezza arm. The treatment difference was 0.31%, and the CI was 0.15% to 0.48%. For this sensitivity analysis, the non-inferiority criterion was not met. Analysis 2 was another analysis submitted by the applicant. This analysis adds 0.4% only for subjects that were adjudicated to be missing not at random. For this analysis, missing not at random was defined as due to apparent lack of efficacy as adjudicated by the applicant. There were only 5 subjects who were clearly identified to have dropped out due to apparent lack of efficacy. Therefore, analysis 2 adds 0.4% for only these 5 subjects. And these results are more similar to the primary analysis. This analysis is subjective in that it relies on adjudication of reasons for dropout. In summary, the degree of missing data for A1C at week 24 raises issues on the reliability and confidence in the results. I will now discuss insulin dosing and titration during the trial. The analysis of the efficacy endpoints cannot be fully interpreted without an examination of the relative doses of insulin used by each study arm. Here we see a comparison of the dosing and titration of basal insulin between the two treatment arms. As plotted in blue on the graph and shown in the upper line, the average daily dose of basal insulin during the trial was higher in the Afrezza arm than in the aspart arm, plotted in black. Additionally, basal insulin appears to have been titrated more aggressively in the Afrezza arm. In an attempt to understand why there was a higher daily dose of basal insulin used in the Afrezza arm, FDA examined several possibilities, including whether perhaps the insulin aspart group was titrated less because more subjects were already at the fasting glucose target. From this figure, we can see that only a small percentage of subjects in each treatment group reached basal insulin titration targets by week 12. This finding suggests that the differing basal insulin use between treatment groups was not due to subjects having reached titration goals. Turning to prandial insulin titration. In order to compare the prandial dose of Afrezza to aspart, we used the conversion algorithm, where 10 units of Afrezza given with the Gen 2 device is approximately equivalent to 4 international units of aspart, which is a ratio of 2.5 to 1. Therefore, to convert Afrezza units to aspart units, we divide the Afrezza units by 2.5 to express them in aspart equivalent units. Here we see a comparison of the dosing and titration of prandial insulin between the two treatment arms. The mean daily doses of prandial insulin increased throughout the study in the Afrezza arm, shown in the blue line. In contrast, in the aspart arm, shown in the black line, the mean daily dose of aspart showed only a slight increase. From this figure, it is apparent that the Afrezza group underwent titration through week 12, as specified in the protocol, but that the aspart group underwent remarkably minimal titration. Therefore, similar to basal insulin titration, we can conclude that higher doses of prandial insulin were used in the Afrezza arm, and more aggressive prandial insulin titration occurred in the Afrezza arm. Please keep in mind that this was in the face of lesser efficacy of Afrezza versus aspart. Again, we explored reasons for why the aspart arm appeared to undergo virtually no titration. From this figure, it is clear that the lack of titration in aspart dose was not because subjects were already at target. There were fewer patients in the aspart group that reached titration targets than in the Afrezza group. This finding suggests that the differing prandial insulin titration between treatment arms was not due to subjects having reached titration goals. And this may be a concern given the open label nature of the trial design. Whatever the reason for the difference, this finding is important because the inadequate titration of aspart raises doubts as to whether we are judging Afrezza against an optimally performing comparator, which is a crucial aspect of a non-inferiority trial. Responder rate analyses examine the percentage of patients who reach glycemic targets recommended by professional societies. In this FDA analysis, we analyzed the responder rates for the subgroup of patients who reached glycemic goals and were above those goals at baseline. Therefore, this subgroup analysis examines the rate of patients who were able to reduce A1C to goals rather than simply maintain their glycemic control. The proportion of subjects who achieved the glycemic goal of 7% or less at the end of the trial was 10% in the Afrezza group versus 21% in the aspart group. Only 5% of Afrezza subjects versus 10% of aspart subjects had an A1C decrease to 6.5% or less. The main points from these data are that overall, the responder analyses are consistent with the analysis of the primary endpoint in that Afrezza appears to be less effective than aspart. In addition, the relatively low responder rates are consistent with the data showing that both groups were inadequately titrated during the trial to reach glycemic goals. So in summary for trial 171, Afrezza was found to be non-inferior to aspart in the primary analysis. However, Afrezza was statistically worse than Aspart. Also, responder rates were better with Aspart versus Afrezza. The caveats to interpretation of the trial results include the high and differential dropout rates, which prompt concern regarding missing data. Non-inferiority was not demonstrated in FDA's sensitivity analysis. Further, non-inferiority studies rely on the assumption that the comparator worked as expected. In this trial, Aspart was minimally titrated, and the average daily basal and prandial insulin doses used in the Afrezza group were consistently higher than those used in the Aspart group, in the face of a lesser improvement in A1C. The next topic for presentation is Study 175, the Phase 3 study in type 2 patients. In Study 175, Afrezza was studied against placebo as add-on to metformin alone or to 2 or more oral diabetes drugs. Subjects were randomized in a 1-to-1 ratio to receive either Afrezza or placebo and were treated for 24 weeks. The protocol included a provision under which subjects with persistent hyperglycemia could receive rescue therapy in addition to their study treatment. The primary study objective was to demonstrate that mealtime Afrezza Gen 2 was superior to placebo in reducing A1C after 24 weeks. This slide summarizes the subject characteristics. I would like to draw your attention to a couple of points that best help characterize the stage of diabetes of the study population. About a quarter were on metformin alone, but the majority of subjects were on 2 or more oral diabetes drugs, and the subjects had a mean duration of diabetes of 9 years, suggesting that overall, these were not recently diagnosed patients. The primary efficacy results for Trial 175 are presented here. The baseline A1C in both groups was 8.3%. The 0.84% mean reduction in A1C from baseline to week 24 in the Afrezza group was statistically significantly greater than the 0.41% mean reduction observed in the placebo group. The treatment difference was 0.42%. The superiority of Afrezza over placebo in reducing A1C was demonstrated since the upper bound of the CI was less than 0, the predefined superiority margin. FDA has no concerns regarding the impact of missing data or data after rescue therapy on the primary efficacy analysis results for this trial. It is interesting, though, that 6.8% of Afrezza-treated subjects required rescue therapy, which is somewhat surprising for a titratable product. To put these results into context, this table shows the A1C reduction achieved in studies of diabetes drugs representing various classes, studied in combination with metformin alone or metformin and another diabetes drug. This table is also included in the backgrounder. I will pause for a moment so you can review it here. While cross-study comparison is generally not recommended, on its face, the magnitude of the effect size achieved with Afrezza in comparison to these other drugs could be considered modest in light of the fact that Afrezza is an insulin and can be titrated, in contrast to the drugs listed in this table. The FDA statistician analyzed the responder rates for the subgroup of patients who reached glycemic goals and were above those goals at baseline. For both glycemic goals, the Afrezza group performed better than placebo and consequently support the primary efficacy analysis. Note, however, that the proportion of subjects reaching less than 7% who were above 7% at baseline with Afrezza was under 1/3. To summarize Trial 175, Afrezza was superior to placebo, both on a background of metformin alone or at least 2 other oral diabetes drugs, with a placebo-adjusted reduction in A1C of 0.42%. Missing data and rescue therapy did not affect the primary efficacy analysis. Responder analyses were consistent with the primary efficacy analysis. The efficacy of Afrezza for type 2 diabetes may be considered modest compared to some of the other available anti-diabetes therapies, including non-insulin oral anti-diabetes drugs. I would like to take a couple of minutes to discuss secondary endpoints of the 2 trials just presented. In the type 1 trial, the fasting glucose decreased from baseline to week 24 in the Afrezza group and increased in the aspart group. Note that this finding is not consistent with the primary efficacy analysis that showed Afrezza is less effective than aspart. This finding may be reflective of the higher basal insulin dose used in the Afrezza arm compared to aspart. In the type 2 trial, there was a greater reduction in fasting glucose with Afrezza versus placebo, but the difference was small and not statistically significant. This finding may be reflective of Afrezza's use as a mealtime insulin, which may not target fasting glucose. Change of body weight was also a secondary endpoint in the trials. In the type 1 trial, Afrezza subjects showed an average weight loss, while aspart subjects showed an average weight gain. In the type 2 trial, the Afrezza subjects showed a slight weight gain, while the placebo subjects showed a slight weight loss. For the type 2 trial, weight gain versus placebo is expected for an insulin. The concept that a less effective insulin would be expected to cause less weight gain is important to consider when interpreting these data. I would like to close by circling back to the beginning of the talk and showing the results of the primary efficacy analyses of older studies and new studies together. For type 1, it is clear that the results of trial 171 are similar to the results of trial 009. In trial 009, the upper bound of the CI was 0.4% in the primary efficacy analysis, just crossing the non-inferiority margin. Although of note, in FDA sensitivity analyses, the upper bound of the margin was higher than the 0.404% that you have seen today. In trial 171, the upper bound of the CI was 0.37% in the primary analysis. However, in the sensitivity analysis, the upper bound was 0.48%. The confidence in the primary efficacy results for trial 171, given the trial conduct issues outlined in this presentation, is one of the topics for discussion today. For type 2, we now have a placebo-controlled trial to consider, in addition to the 2 active comparator trials. In trial 175, Afrezza was superior to placebo. In clinical practice, however, the decision facing providers is not to treat with Afrezza or placebo, but rather Afrezza or another drug. A mean reduction in A1C of 0.42% could be considered modest for an insulin. Afrezza's role in the armamentarium of options to treat type 2 diabetes is a topic for which we would appreciate the committee's input. Thank you.
Good morning. My name is Mia Paterniti, and I'm an allergist immunologist and clinical reviewer in the Division of Pulmonary Allergy and Rheumatology Products, known as DPARP. The Division of Metabolic and Endocrine Products, or DMEP, has requested consultation from DPARP on multiple occasions throughout the Afrezza review process to evaluate pulmonary safety given the inhaled route of delivery. Over the next 20 minutes, I will summarize the pulmonary safety data for Afrezza. This is an outline of my presentation. I will start by providing background and regulatory history for the application. Next, I will present the pulmonary safety data submitted in the original submission. I will follow this by detailing the pulmonary safety data for the 2 clinical studies in the resubmission. I will close with some summary and concluding remarks. Before moving into the pulmonary safety findings for Afrezza, it is important to consider both the drug and the device through which it is being delivered. The drug is Technosphere insulin, or TI. I will discuss the device in subsequent slides. As outlined previously by the sponsor, TI is a dry powder formulation of recombinant human insulin. It uses a novel excipient, fumarate diketopiperazine, or FDKP. FDKP self-assembles into Technosphere particles under acidic conditions. The drug product is formed by adsorbing insulin onto the preformed technosphere particles. The particles dissolve under physiologic conditions, releasing the adsorbed insulin. As outlined previously by Dr. Yanoff, the Afrezza new drug application was originally submitted in March 2009. The proposed product at that time was TI delivered through the Medtone inhaler. The original submission included 9 controlled trials spanning 11 weeks to 2 years in duration. The applicant received a complete response from the FDA due to efficacy and device issues. In order to address the deficiencies outlined by the agency, the application was resubmitted in June of 2010 with a new device, the Gen 2 inhaler, used to administer the same drug product. In this resubmission, the applicant provided data to bridge the Medtone and Gen 2 inhalers through in vitro and clinical pharmacology studies. No clinical data was provided to assess the efficacy and safety of the new Gen 2 device. The applicant received another complete response from the FDA requesting clinical trial data with the new Gen 2 inhaler to support its efficacy and safety. In October of 2013, in their second and current resubmission, the applicant conducted 2 new phase 3 clinical trials of 6 months duration in which TI was delivered via the Gen 2 inhaler. The primary objective for these studies was efficacy. However, descriptive pulmonary safety data was also obtained with the new Gen 2 device. Because no substantial pulmonary safety data was provided in the first submission, the remainder of the presentation will focus on the original submission and resubmission number 2. Seen here are the Medtone and Gen 2 inhalers. The Medtone inhaler was the original device used to deliver TI. The Gen 2 inhaler is the to-be marketed device. This table compares the Medtone and Gen 2 inhalers. Both devices are breath-actuated dry powder inhalers that use the same novel excipient as a carrier for insulin. The Gen 2 inhaler is proposed to be a more efficient delivery system, requiring less insulin per cartridge. The 2 devices also differ in administration, cleaning, and the use period. As I go through this presentation, I will be comparing the pulmonary safety data obtained in the original submission, in which TI was delivered via the Medtone inhaler, to the current data, in which TI was administered via the Gen 2 inhaler. It is important to keep in mind the device changes when evaluating the pulmonary safety data, as pulmonary safety effects are local and can be affected by substantial changes in device. Having provided the relevant background information, I will next turn to a discussion of the pulmonary safety data from the original submission. The original submission included 9 controlled studies in type 1 diabetes, 6 controlled studies in type 2 diabetes, and 1 study in both type 1 and type 2 diabetes. Adverse events were analyzed from a pooled safety population incorporating all of these studies. Pulmonary function was assessed in study 009, the 1-year study in type 1 diabetes, study 102, the 1-year study in type 2 diabetes, and study 030, the 2-year study in type 1 and type 2 diabetes. This table shows the number of subjects enrolled by treatment arm and disease type. Approximately 2,400 subjects were treated with Afrezza, 2,000 subjects were treated with a non-inhaled comparator, and 100 subjects with the inhaled excipient technosphere particles without insulin. Subjects were excluded from the phase 3 clinical studies if they had COPD, asthma, or other significant pulmonary disease, were current smokers or had a smoking history within the past 6 months, had a history of malignancy within the past 5 years, or had abnormal lung function defined via spirometry. 3 pulmonary safety findings were identified during the review process: FEV1 decline over time, bronchospasm in patients with asthma and COPD, and cough. Given the importance of FEV1 measurements, it may be helpful to review the measurement of lung function, or spirometry. Spirometry testing measures various aspects of breathing and lung function. Two measurements obtained through spirometry are particularly useful: FEV1, or forced expiratory volume in 1 second, and FVC, which is the forced vital capacity. FEV1 is frequently used as a primary endpoint for many pulmonary trials. The best of 3 acceptable and reproducible efforts are used to obtain these measurements. The lower limit of normal for FEV1 and FVC is 80% of the predicted value. The clinical studies for Afrezza excluded subjects with FEV1 or FVC less than 70 to 80% predicted, depending on the study. As the measurements for FEV1 and FVC showed similar trends, I will focus my discussion here today on FEV1. This table shows the mean change in FEV1 from baseline in type 1 diabetics at 6 months, 1 year, and 2 years. Comparing Afrezza to the non-inhaled comparator at each time point, the treatment difference was 0.04 liters. This treatment difference demonstrates that patients treated with Afrezza delivered via the Medtone inhaler had a small but greater decline in FEV1 compared to patients treated with the non-inhaled comparator. While the difference of 0.04 liters itself is not likely to be clinically relevant, effects of continued chronic administration beyond 2 years are unknown. This figure shows the mean change in FEV1 from baseline over 2 years in type 1 diabetics. The Afrezza arm is represented by the lower line, and the non-inhaled comparator is represented by the upper line. The vertical line marks the 3-month time point. As displayed, the decreases in FEV1 occurred in the first 3 months and persisted over 2 years. The difference did not appear to be progressive, although we do not have controlled data beyond 2 years. There was insufficient data to draw definitive conclusions regarding reversal of the FEV1 due to few patients contributing follow-up data after withdrawal of treatment. The nature and magnitude of the change in FEV1 was consistent with what has been observed in Exubra, another inhaled insulin. Studies in type 2 diabetics showed similar values. Since patients with asthma or COPD were not included in the phase 3 clinical studies, the applicant completed 3 small studies in patients with asthma or COPD, with 1 ongoing study as listed in this table. In the completed study, a total of 22 patients with asthma and 18 patients with COPD were studied. Of the 18 patients with COPD, only 8 had measurements of pulmonary function. The ongoing study, Study 134, has enrolled only 17 subjects since enrollment began in 2007. In this study, 2 of 4 patients with COPD randomized to TI have discontinued due to pulmonary exacerbation. I will discuss the results of Studies 113 and Study 015 in more detail on the next slide. A total of 22 patients with asthma were enrolled in clinical studies. One of these studies with 17 asthmatics showed a clinically significant mean decline in FEV1 post-Afrezza of about 400 mLs, with a return towards baseline FEV1 by 2 hours. 2 serious adverse events of bronchospasm were reported. Both were for wheezing 15 to 30 minutes post-Afrezza inhalation with drops in FEV1 of 33 to 45%. Both subjects' symptoms resolved with beta agonist treatment. Bronchospasm and wheezing adverse events occurred in 29% of asthmatics compared to no events in non-asthmatics. One COPD study showed a smaller mean decrease in FEV1 post-Afrezza of about 200 mLs in 8 COPD patients. FEV1 returned towards baseline by 8 hours. There were no serious adverse events. Respiratory AEs were similar for both patient populations. The acute bronchospasm seen in these clinical studies is clinically significant. In the original submission, after hypoglycemia, cough was the most common adverse event noted in the pooled safety population, occurring in 27% of patients. Cough usually occurred within 10 minutes, was generally mild, dry, intermittent, and tended to decrease over time. Cough was also the most common reason for discontinuation due to an adverse event occurring in about 3% of patients. The frequency of cough was similar to what was seen in the Exuber program, but more than what is typically observed for dry powder inhalers developed for the treatment of asthma or COPD. A summary of the respiratory serious adverse events for the original submission are listed in this table. This table does not include patients with asthma or COPD. Overall, the incidence of respiratory serious adverse events in the overall safety population was rare. In general, bronchospasm was not a major issue. Up until this time, the data I have shown you was generated in the original program with TI delivered via the Medtone inhaler. Using this discussion as a foundation, next, we turn to the resubmission, which included pulmonary safety data from 2 clinical studies using the Gen 2 inhaler. This table outlines the 2 6-month clinical studies included in the resubmission. With respect to pulmonary safety, Study 171 compared TI delivered via the Gen 2 inhaler to TI delivered via the Medtone inhaler in type 1 diabetics. Study 175 compared TI to the excipient, technosphere particles without insulin, or TP, both delivered in the Gen 2 inhaler. The number of subjects enrolled in these 2 studies comprised about 12% of all TI-treated subjects. The primary objective for both was efficacy. However, FEV1 measurements were also included. Pulmonary exclusion criteria were similar to the original submission. Note that the 2 studies only provide 6 months of pulmonary safety data with the Gen 2 inhaler as compared to 2 years with the Medtone inhaler. This table shows the mean change in FEV1 in liters from baseline in type 1 diabetics at 6 months in Study 171 compared to the original submission. There was no significant difference in the mean change from baseline in FEV1 between the TI Gen 2 and the TI Medtone groups at 6 months. Although no analysis was done in Study 171 to compare the TI Gen 2 or TI Medtone arms to the comparator, the numerical differences of 0.03 liters for the Gen 2 inhaler and 0.04 liters for the Medtone inhaler are similar to the original submission. This table shows the mean change in FEV1 in liters from baseline in type 2 diabetics at 6 months in Study 175 compared to the original submission. The change in FEV1 from baseline in TI compared to the inhaled excipient was similar for both the Gen 2 and Medtone inhalers. Similar to the original submission, the 2 studies with the Gen 2 device also reported cough as the most common respiratory adverse event, with similar incidence rates and early discontinuation rates compared to the original submission as seen in this table. Cough characteristics were also similar. Notably, in Study 175, the inhaled excipient had similar cough rates to TI, suggesting that the novel excipient, FDKP, may be playing a role in respiratory tract irritation. A similar finding was also seen in the original submission. 1 SAE of bronchial hyperreactivity was reported in Study 171 in the TI Gen 2 treatment group. Overall, the pulmonary safety data obtained with the Gen 2 inhaler was similar to the Medtone inhaler. I will now close with summary and concluding remarks. In summary, the pulmonary safety data in the original submission spanned to 2 years, up to 2 years, with TI delivered via the Medtone inhaler. Subjects were excluded for underlying lung disease, smoking, and abnormal lung function. The mean FEV1 treatment difference over time between Afrezza and the non-inhaled comparator was small, about 40 to 60 mL. The treatment difference was noted within the first 3 months and persisted to 2 years, but did not appear to be progressive. In the small asthma and COPD studies, patients with asthma had a post-inhalation FEV1 decline of approximately 400 mLs. A smaller decline was noted in COPD patients of 200 mLs. Bronchospasm and wheezing was noted in 29% of patients with asthma. Cough was the most common adverse event and the most common reason for discontinuation. The applicant proposed a new device, the Gen 2 inhaler, to address issues identified during the review of the original submission. 2 controlled clinical trials up to 6 months in duration with the Gen 2 inhaler were conducted. The pulmonary safety profile for TI delivered via the Gen 2 inhaler was similar to TI delivered via the Medtone inhaler. However, the number of patients in the clinical program with the Gen 2 inhaler was smaller and the duration of treatment shorter. From a pulmonary safety standpoint, a key issue in the Afrezza clinical development program is the significant change in device from the Medtone to the Gen 2 inhaler, because pulmonary safety is local and may be affected by a change in device. In addition, it is important to consider the shorter duration and the smaller number of subjects treated with TI via the Gen 2 device when evaluating the data submitted. With the change in device and the potential limitations of the Gen 2 data in mind, we ask you to consider the pulmonary safety data with the Gen 2 inhaler are sufficient to support the pulmonary safety of Afrezza. Having completed the pulmonary safety discussion, I will now turn the podium over to Dr. Lisa Yanoff, who will discuss the non-pulmonary safety of Afrezza.
Hi again, I'm Dr. Yanoff, clinical reviewer in DMEP, and the focus of my second presentation today is the non-pulmonary safety of Afrezza. For the non-pulmonary safety evaluation, I will focus specifically on the topics of lung cancer, diabetic ketoacidosis, abbreviated DKA, and hypoglycemia. I will end with some closing remarks summarizing the non-pulmonary safety issues that FDA would like the committee to consider when discussing the risk-benefit profile of Afrezza this afternoon. This afternoon, the committee will be asked to discuss the potential lung cancer signal for Afrezza. Given the mode of administration of Afrezza and the experience with the inhaled insulin Exubera, which will be discussed in the next presentation, lung and bronchial cancers are a safety concern. This concern also stems from the notion that human insulin may have potential mitogenic properties via IGF-1 receptor binding. While CAR-C studies were negative for Afrezza, there are limitations to the methods used in the non-clinical studies. Further, the existing data do not address whether Afrezza may promote or enhance pre-existing premalignant bronchial or lung lesions. In the Afrezza development program, 4 cases of lung cancer were reported. 2 in the original NDA that are shown in the turquoise shaded top half of the table, and 2 cases that were spontaneously reported after the subjects had completed participation in the clinical trials. These are shown in the gray shaded bottom half of the table. The 2 patients diagnosed during the study had a prior history of heavy tobacco exposure and so were at higher baseline risk for lung cancer. The 2 spontaneously reported events of squamous cell lung cancer are of particular concern because the patients were reported to have no history of tobacco use or other known risk factors. The detailed narratives for all the cases of lung cancer are located in the briefing document. The incidence of lung cancer among Afrezza-treated patients can be calculated in different ways depending on the denominator population. In the pooled Phase 2-3 safety database, there was 1 reported case of lung cancer, and the exposure-adjusted incidence rate of lung cancer was about 50 per 100,000 subject years of exposure. In a database that includes all Phase 2 and 3 controlled and uncontrolled studies of greater than 14 days duration, The exposure-adjusted incidence rate of lung cancer was 73 per 100,000 subject years of exposure, because of the additional case of the patient in the uncontrolled study 010. For the entire Afrezza exposed database, we observed an imbalance of 4 cases for Afrezza and none for comparator. Interpretation of this 4-to-0 imbalance should be taken with caution because it includes 2 spontaneously reported cases. Moving on to DKA. During the original NDA review, we noted an imbalance of events with MEDRA preferred terms indicating DKA of 13-to-3 not favoring Afrezza. There were no new cases of DKA in the resubmission, although note there was only 1 new trial in type 1 patients in the resubmission. The 13-to-3 imbalance translates into about a 4 to 5-fold higher risk of DKA for Afrezza subjects versus comparator subjects based on exposure-adjusted estimates. Review of narratives were able to identify some triggers, including viral or bacterial infection or noncompliance with insulin therapy. But caution should be taken in trying to explain away the imbalance as primarily due to infections or other triggers, because randomization should have balanced treatment groups for predisposing factors. In light of the observed lesser efficacy of Afrezza versus Aspart in type 1 patients, the possibility exists that Afrezza contributed to this observed imbalance in DKA. Regarding hypoglycemia, intensive review of hypoglycemia data from the original NDA showed that the incidence of hypoglycemia was higher with Afrezza than placebo in placebo-controlled studies, which is expected based on Afrezza's mode of action. Also, the incidence of hypoglycemia was generally lower for Afrezza than for comparator insulins, but in these studies, most of the comparator groups had better glycemic efficacy than Afrezza, confounding the hypoglycemia analyses. Therefore, FDA concluded that there was no clear, consistent evidence of hypoglycemia benefit of Afrezza. For the new data in the resubmission, the results appear consistent with the original NDA. In the type 1 diabetes trial against insulin aspart, the rate of both severe and all hypoglycemia was lower for Afrezza. However, rather than concluding a hypoglycemia benefit, we believe these results may reflect the finding that Afrezza was less effective than comparator in the trial. In the type 2 diabetes trial, 175 against placebo, the rate of both severe and all hypoglycemia was higher for Afrezza, which is expected for an insulin. Similar to the original NDA review, we conclude that there is no clear, consistent evidence of a hypoglycemia advantage of Afrezza. In summary, for potential lung cancer signal in the Afrezza program, we observed a numeric imbalance of 4 cases to 0. We would like the committee to discuss the level of concern regarding these data. Further information about the risk of lung cancer in the SEER database and for the inhaled insulin Exubera will be given in the next FDA presentation. For the safety issue of DKA, we observed an imbalance of events of DKA not favoring Afrezza. Hypoglycemia does not appear to be a safety concern for Afrezza above and beyond what is typically observed for other insulins. However, we also do not believe that a hypoglycemia advantage for Afrezza has been clearly established. Taken together, the higher rate of DKA and the lower rate of hypoglycemia observed for Afrezza may be indicative of its lesser efficacy. We would like the committee to consider this possibility in evaluating the risk-benefit profile of Afrezza.
Hello, so I'm Patricia Bright, and I'm an epidemiologist with the FDA. And the objectives of my talk are to provide background on the Exubera experience, another inhaled insulin, to help inform decisions about Afrezza, to share information presented by Pfizer representatives at the International Conference on Pharmacoepidemiology in August 2012. And this covered the follow-up study of the Exubera clinical trial population, known as FUSE, for lung cancer incidence, lung cancer mortality, and all-cause mortality. And I'll be discussing the limitations of post-marketing strategies to evaluate a possible lung cancer risk with Afrezza. So as background, Exubera is an orally inhaled insulin from Pfizer. It was approved by the FDA in January of 2006 for the treatment of adults with type 1 and 2 diabetes. The labeling stated that it was contraindicated in smokers and those who had discontinued smoking less than 6 months. There was an identified risk of small, non-progressive, and reversible decrease in pulmonary function. There was also a hypothetical risk of serious pulmonary events and lung cancer. The product labeling was changed in September 2008 to reflect lung cancer cases with Exubera use. It was also withdrawn by the sponsor in September 2008 due to lower than expected sales. So now I'm presenting some drug utilization data. In blue you have the prescriptions, in red you have the patients, and you have time on the horizontal axis. Marketing started in the second quarter of 2006. It peaked in the third quarter of 2007 and it declined in 2008. And you'll see I put a purple arrow in here to indicate when Pfizer first publicly announced it would stop selling Exubera. This was October of 2007. I also included an arrow to indicate when the labeling change took place in September of 2008. The slides I'm going to be showing you were copied and adapted for this presentation, and this is the source. So there was a known imbalance of lung cancer cases at the NDA filing. By 2008, there were 5 in the Exubera exposed and 1 in the non-exposed. This gave a crude hazard ratio of 5.1. All cases were among former smokers. There was possible detection bias for lung cancer because most patients were randomized in open-labeled, unblinded trials. There were mild pulmonary symptoms associated with Exubera use, such as the cough we heard about, and there was a novel route of insulin administration. All of these could have led to more frequent and more thorough pulmonary assessments. The sponsor was interested in investigating this imbalance further, so they conducted the follow-up study of the Exubera clinical trial population, known as FUSE. The aim of the FUSE study is to evaluate whether Exubera-exposed patients experience primary lung cancer mortality at a higher rate than the unexposed patients. The primary endpoint was primary lung cancer mortality. Secondary endpoints were primary lung cancer mortality among former smokers, primary lung cancer incidence, and all-cause mortality. Lung cancers and lung cancer deaths were reported by study investigators. They were adjudicated by an external expert committee that was blinded to randomized group. And we'll be discussing the lung cancer cases classified as highly likely or likely. Study design followed the participants from the original 17 clinical trials. These were conducted at 320 sites in 25 countries. The idea was to follow them forward prospectively without any study interventions to look for endpoints. But the clinical trials stopped at different times, so sometimes there was an interim period between the end of a clinical trial and the start of the prospective follow-up. For those sites that were participating in the prospective follow-up, we were able to obtain data during this interim period. All of the data points together contribute to the FUSE data. The 2012 Pfizer presentation included results for in-study, which did not include the interim data, but they also presented the continuous data, which did include the interim. And here we're presenting the continuous data. So if you look at the oval in the top left-hand corner, those are your participants from the original clinical trials. There's 7,439. Some of the sites chose not to participate in the prospective follow-up portion. That automatically eliminated 3,092 patients. Of those sites that did participate, some individual participants chose not to participate in the prospective portion. So we had another 1,716 drop out at that point. That left 2,631 patients that entered the FUSE prospective portion of the study. Only 95 did not complete that prospective portion. This left us with 34% of the original clinical trial participants completing the prospective portion of FUSE. At baseline, potential confounders were measured from the original trial. It's included age, gender, race, smoking, pack years, body mass index, hemoglobin A1C, lung function tests, type of hemoglobin, history of cancer and asthma, and COPD. They were balanced across all exuberant and comparator subjects, exuberant and comparator subjects who participated in prospective follow-up, and the prospective participants and the non-participants. Additional covariates were measured at the fused baseline, and these were balanced across exuberant and comparator subjects. This is just an example of the smoking history. This is for the prospective subjects only, but you can see by smoking history they had a balance in never smoking between the exuberant exposed and the comparator. And if you look at the bottom, you'll see the quartiles for pack years of smoking, and this is relatively balanced as well. Now we're looking at the endpoints. The top row here is primary lung cancer incidence. The middle row is primary lung cancer mortality, and the bottom is all-cause mortality. I'm going to have you focus here on primary lung cancer incidence first, and the reason is the mortality data is actually rolled into this as incident cancers. And in particular, I want you to look here in the red box at the highly likely or likely cancers. You'll see that there's 12 in the exuberant exposed and 3 in the comparator. It's this imbalance that's driving the concern for lung cancer. You would expect some more cases in the exuberant exposed with detection bias because they're going to be experiencing the coughing, and it's a novel route of administration. But it's the magnitude of the imbalance which is causing concern. I also want you to look over here in the blue box. This is among former smokers, and you'll see that 14 of the 15 cases were in former smokers. I do want to point out this is not out of line with the risk that you would expect from smoking alone, but it did raise the possibility that there could be a promotional effect between exuberant and the former smokers. And last, I'd like you to look down here at all-cause mortality because the imbalance actually goes the other way, and you see more deaths in the comparator group. So now we're going to look at the risk estimates. If you look at the lung cancer incidence, had an elevated incident density ratio of 3.75, and this was statistically significant. Below it, you have lung cancer mortality, also elevated at 2.81, but not statistically significant. And all-cause mortality appeared to be protective, but was not statistically significant. Now I'm going to show you the timing. 4 of the cases occurred in the original clinical trials, which is the arrow on the left. 3 of the cases occurred in the prospective follow-up, which is on the right, and 8 of the cases occurred during the interim period. This is the same slide. I've just added the person years now as the denominator for people to see. So, study strengths. It's a large source population with baseline trial data available for patients, allows for comparisons of baseline trial data among participants and non-participants, exuberant and comparator prospective fused participants. It's geographically and ethnically diverse study population, although I would like to point out that only 7% of participants were black. And it was balanced in all measured potential confounders at baseline. Study limitations. It had low participation of sites and patients in the prospective portion, so only 34% of the original trial participants completed the fused prospective follow-up. There was a potential for detection and reporting bias in endpoints for the exuberant exposed, and there's possible changes over time in covariates. There were small numbers leading to insufficient power to draw clear conclusions. This is Pfizer's summary that the FDA agrees with. The data were indicative of a potential increased lung cancer risk, but they were inconclusive. There was a trend toward increased risk may be explained by reporting or detection bias associated with preferential screening and reporting of cases for subjects exposed to exuberant in the original studies, a promotional effect of exuberant among smokers, or a combination of both. And there was no increased risk in all-cause mortality. Now we're going to discuss the post-marketing assessment of Afrezza. We just saw that Exubera was an orally inhaled insulin with a potential risk for increased lung cancer, but inconclusive data. So how do we evaluate for an increased lung cancer risk with Afrezza? I'll be presenting strategies for post-marketing assessment of lung cancer risk, and we'll discuss 3 approaches. But I want to forewarn you that there's a lot of challenges in doing this, so you're going to see a lot more limitations than you do strengths. So here's one of our challenges. The comparison groups may not be similar. Users of standard insulins may differ from users of inhaled insulins. We have small expected numbers of outcomes, and this is going to be an issue with almost every approach because this will affect feasibility. A long follow-up time is needed for a latency period. Drug uptake is unknown. Mean length of exposure to Afrezza is unknown. There could be detection bias in the Afrezza exposed. And smoking is a strong confounder for lung cancer. So first I'm going to discuss the sponsor-proposed approach. They suggest a prospective observational voluntary product exposure registry without an internal comparator. The exposure would be Afrezza use. The primary outcome is incidence of primary pulmonary malignancies in patients taking Afrezza. Secondary outcomes are all other malignancies, except non-melanoma skin cancers, serious pulmonary allergic and hypoglycemic events requiring medical intervention. They suggest use of an external comparator, which would be the incident rate in the U.S. population based on SEER data. They suggest 200 sites with patients both type 1 and type 2 diabetes in the U.S. with other countries added. Participants age 18 and over who are starting Afrezza and who have provided informed consent. Would be non-interventional with usual care visits at a minimum of every 6 months. With 1,800 patients enrolled and followed for 5 to 7 years, and comparing lung cancer incident rate in the registry to incident rate in the U.S. population based on age-adjusted SEER data. They suggest that if the lower limit of the 95% confidence interval is above 64.6 per 100,000, then exposure to the Afrezza would be deemed a significant risk for pulmonary malignancies. So a strength is this may be the quickest approach because all participants would be invited and they would all go into one arm of a study, but there are limitations. It's voluntary and it's unknown enrollment. You'd have detection bias, which will increase the rate in the registry even if Afrezza does not increase risk. Inhaled insulin users differ from the general population. And there's unmeasured smoking in the SEER population, so you're going to get confounding. And also limited covariate collection in the SEER data, which may lead to more unmeasured confounding. And I just want to show you the incidence rates for pulmonary malignancy. In the general U.S. population, according to age-adjusted SEER data, it's 61.4 cases per 100,000 person years. In the Exubera plus Fuse data that I just showed you, it was 107 per 100,000 person years. In the Afrezza trial, based on just the 2 cases and not the spontaneous reports, it would be 73 per 100,000 person years. But I do want to mention that the lung cancer incidence is strongly driven by smoking history in the population. And from what I've generally seen in diabetic populations, they tend to be much higher than this. So I will also show you the option of a registry with an internal comparator. This could be set up similarly, 200 sites, patients with type 1 and 2 diabetes in the US and other countries added. Participants age 18 and over starting Afrezza. And the comparison group would be defined by exposure duration. So it would be, the exposure would be categorical, short versus longer Afrezza duration. And basically what we would want to do here would be to catch people who stop the drug right away after starting it and use them to compare the people who stay on the Afrezza. The outcomes, this is sort of a placeholder because it would actually have to match whatever, if we issue a post-marketing requirement, it would have to match what that says. But we have here incidence of lung cancer mortality and all-cause mortality at 3, 5, and 10 years by Afrezza use. This approach would allow for careful documentation of confounders and adjustment for pack years of smoking. Strength, you'd have increased similarity between the comparison groups. But again, there's limitations. It's voluntary, so unknown enrollment. Those stopping Afrezza could be different from those continuing in terms of side effects or adherence differences. You still might have detection bias with coughing on the Afrezza group for those who continue on. And it might require larger sample sizes and take a longer time to accumulate the registry participants because now you have the participants, the Afrezza users feeding into 2 arms of a trial. And the cut points for shorter use versus longer use would have to be determined. Another approach would be a randomized clinical trial. The exposure could be randomized to Afrezza use versus standard of care. Outcome, again, this is a placeholder. It would depend on the PMR, but what we're saying here is incidence of lung cancer mortality, and all-cause mortality, adjusting for pack years of smoking. Same general parameters in terms of 200 sites, patients with type 1 and 2 diabetes in the U.S. with other countries added, age 18 and over starting Afrezza with informed consent. Strength, the randomization would reduce bias due to unmeasured confounding. However, again, we have some limitations. Patients may refuse enrollment, especially if they have needle phobias. You still have the detection bias with higher rates likely in the Afrezza group. And notably, that given the small numbers, to detect a relative risk of 2 or more, the sample size would need to be 31,000 per group. This is an issue with any of the studies. So in summary, evaluating lung cancer risk with inhaled insulin use in the post-marketing setting is challenging. Several approaches may be needed to assess risk. Each has advantages and disadvantages, but smoking data are critical in assessing the lung cancer risk. And thank you.
So we now have, thank you to the FDA, and we now have time for some questions related to clarification. I'd like to focus on the FDA presentation. Start with David Cook.
So my question was related to the FDA's interpretation that there was no hypoglycemia benefit clearly demonstrated, and maybe explicitly for me and us, commenting on the sponsor's slide CE39 that showed lower hypoglycemia with each of the different A1C outcomes, and why you don't think that that's supportive of the lower hypoglycemia?
So I think some of the issues were brought up before. I think one of the issue was the post hoc nature of these analyses. So if you look at CE39, this is basically on post hoc data. It's based on patients who actually, I think, made it to week 24. So you'd have to consider the large differential dropouts in both groups, and you're basically assuming that the two groups at week 24 are the same so that you can compare them head to head. We're not sure that's possible. The other thing is it's actually comparing event rate and not proportion, although we'd like to see proportion when someone wants to make a hypoglycemia benefit claim. So we'd want to see proportion tracking with event rate, and I'm not sure we've seen those data. And finally, the hypoglycemia is a very difficult thing to capture in a clinical trial. It really relies on the patient to be able to capture the event, and so it's not something like hemoglobin A1C that is centrally measured. It's something that you're basically relying on diary data to make a claim. It's been very, very challenging for any sponsor to claim a hypoglycemia benefit with any real product, and if they were going to do that, we would ask them to design a trial specifically addressing that question. So I think for all these reasons, they're interesting, they're hypothesis generating, but the first thing that we would like to see is really a matched in hemoglobin A1C before we make any sort of declare victory with regards to hypoglycemia. So if the hemoglobin A1C had been really centered on 0 and not on 0.2, we would maybe look at this differently.
Dr. Thomas.
Abraham Thomas. This is a question for Dr. Jain. In your presentation about the coefficient of variation, I just had a question on that. I think for an individual patient, the within-subject variation is really what's important in terms of treatment. You mentioned that some of the variation may be related to multiple inhalations, and I may have missed it in the reading, but is there actual data to show that? I mean, it would be easy to do. You could do the same dose, one with one inhalation and one with multiple inhalations to see what the CV is.
Location FDA. In that particular trial, for lower doses, 10 units and 30 units, patient required only 1 or 2 cartridges, but for higher doses, I think 80 units, they required 4 cartridges. And I'm not aware of the other data. Perhaps the sponsor can answer if they have other data to address that.
Because I would think maybe in early development, there might have been an analysis. You could do exactly that. You could give the 20-unit dose and then to the same patient do 2 10-unit doses and then you would see the effect of inhalation on the CV to see if there's a change, even though the dose is the same.
I believe there was a trial in earlier development comparing 10 units versus 120 units, and perhaps sponsor can add the data, but I would I think I will reserve my point for afterwards. Sorry.
Dr. Swenson.
Yes, I have several questions, and they all relate to issues around the airway effects of the inhaled Afrezza. And very, very quickly, I want to just ask, does the company know whether something like mannitol or methacholine responsiveness changes in subjects that use this compound? It was mentioned that, for instance, pulmonary function studies don't necessarily always pick up asthmatic patients, but given the fact that there's cough as a reaction, could there be some subtle bronchial hyperresponsiveness developing in these patients, which could have several manifestations or implications? And one would be that perhaps subsequent dosing of these different units might not be as efficient. If there's peripheral small airways constriction, there might be dosing that's deposited higher up in the airways. This might lead to some of the questions around differential or non-proportionality in dosing versus both PK and more importantly, PD. So the questions I have are, have you taken the aspects of airway reactivity and airway changes with the compound beyond just looking at pulmonary function data as you've presented?
Mr. Chairman?
Yeah, if you could make just a brief answer, because I want to keep focused on clarification questions for FDA at this point as much as possible. We'll have more time for sponsor discussion this afternoon.
My apologies. I realize. I'm sorry.
That's all right. That's okay.
Would you like me to delay the response?
Yeah, we'll come back to it, and then we'll make sure I don't skip that, Dr. Swenson, as well. Dr. Calhoun.
Thanks, Bill Calhoun. I have two questions for Dr. Yanov and one for Dr. Paterniti. First question for Dr. Yanov. You also commented on your slide 22 that this was an unblinded study. It's a non-inferiority, and so errors in or limitations in the execution of the study will bias toward no difference, which in fact, as we heard this morning, then favors finding a non-inferiority finding, as opposed to a superiority study in which the bias toward the null is a conservative approach. The question is, the FDA has been working with the company for 5 years by my count in pulling this product to market. And so the question is, was the agency responsible for recommending a non-inferiority, unblinded design for the primary efficacy study?
Yes, FDA was involved, me personally, after the first submission, in recommending the trial designs for the two studies that we wanted them to come in with the two new devices. And since we wanted to try to limit it to two studies, one in type 1 and type 2, because we already had some data with the Medtone device, we really had to go for what would be the best trial for each type of diabetes. And in type 1, a placebo-controlled trial would be non-ethical. So,
and explain to me why that is, because that would be an injection. The only thing it would add would be an injection of placebo in those people who are randomized to active inhaled drug, and the addition of a dummy inhaler to those who are getting injected. And that's certainly not, in my view, that's certainly not unethical. We have trials that are far more invasive than that in other therapeutic areas. So I don't think that's unethical at all.
Well, I appreciate your view. In general, in the division of in DMEP, we've approached all, tried to be fair to all the programs, and in general, our approach has been to not require blinded studies for type 1. to require dummy injections.
Second question for you is, when you were talking about the safety, you were concerned about only 6 months duration of safety with the new device, whereas there's 2 years of safety data with the Medtone device. And the question is there a biologically compelling reason for you to be concerned that the device, as opposed to the product, would have a measurable effect on safety? I don't think of one myself, that there's a biologically plausible reason to believe that changing the nebulizer, for example, substantively changes the delivery of albuterol, or changing the delivery device will substantively affect either pulmonary or non-pulmonary safety. So I'm just interested in hearing why the agency raised that as a concern.
Mia Paternity, FDA. The reason we raise it as a concern is it's just an unknown factor in terms of the subtle changes in the device that we just don't have data after 6 months. So we cannot definitively make conclusions similarly to the Medtone in terms of the time and duration. So we're just potentially that there could be some subtleties from the device changes that could come out if it was studied for 2 years. So there's no specific concern and no specific biological plausibility that we identified, but we are putting it out there as a point of discussion.
Last question for Dr. Paternity then is on your slide 22. As I understood your presentation, you basically gave the product a pass for type 2 diabetes, but I noted that there's a 90 ml decrement, differential decrement between excipient and TI, which looks to be, at least if I'm reading the confidence intervals properly, looks to be statistically significant. So a 90 ml decline in FEV1 in type 2 diabetics, which occurs differentially in those who are exposed to adsorbed insulin versus the particles itself, looks to me actually to be something I'd be interested in following up more. What was your thought on that?
Mia Paternity, FDA. Dr. Calhoun, I appreciate you bringing up this point because we also noticed that. And I think if you look at each individual arm, if you look at the TI Gen 2 and the TI Medtone groups, they were actually very similar, so almost numerically identical at 0.13 decline in FEV1 by 6 months. It was actually the comparator group where we saw the difference and why the treatment difference was larger. So if you look at the inhaled excipient using the Gen 2 in study 175, that was actually a smaller decline compared to the original submission data, and that's where the difference, why there is a numerical difference in the treatment difference. So thank you for pointing that out.
Dr. Britton?
Yeah, I want to go back a little bit to the discussion about the delta of 0.4. I mean, the non-inferiority margin of 0.4 that we discussed this morning and mentioned again now as being part of the design. I guess I have a couple of questions about it. One, I think you said you used, and first of all, I want to have you confirm that you use 0.4 in any comparison of insulin, a more traditional insulin versus one like this that might have some real advantages that a traditional one wouldn't have. You could imagine having made a different choice for the margin. No, that's number one. And number two, I'm wondering how good that M1 value of 0.8 is, because I would think it would be very hard to estimate that, and I wonder if it was hard to estimate, and because it was hard to estimate, you made a very conservative choice about what the M1 is. Can you speak to that?
So I think we want to refrain from talking about margins with regards to insulin, but I'll say a few words about the margin. So the way that, so in terms of estimating the margin, the M1 margin, I think that you can estimate an M1 margin for type 2 diabetes. If you were going to estimate an M1 margin for type 1 diabetes, you'd have to look at really the example I gave in my talk, where you basically have everyone on basal insulin, and then somehow you would pass an ethics committee to say that it would be ethical to get a placebo response in type 1. That being said, I'm sure a lot of endocrinologists on the panel will tell you that if you don't have prandial insulin and you only have basal insulin, there's going to be a large differential in HbA1c. And so I'm not really sure the 0.4 is all that conservative. We don't really tell the sponsor what margin is appropriate for their product. When they come to us and they design their program, they have to somehow justify their margin. And the 0.3 to 0.4 is suggested in our guidance document, but it's just a benchmark. We sort of look to sponsor to justify their margin when they're negotiating their protocols with us.
Dr. Stoller.
So my question is to the agency regarding prior experience with Exubera. Falls a little bit outside of the lung cancer discussion, but is germane to an issue of the undetected burden of COPD. So by way of background, I think we're all aware that the prevalence of COPD in this country is probably equal to that of diabetes, but most of it is unrecognized. And we've seen some signals here, as we had seen in Exubera. I happen to sit on that committee as well for the agency, that suggested an accelerated decline and adverse effects in COPD. So my question is, Exubera, like this drug, proposes doing pulmonary function tests as a precondition of prescribing the drug given the adverse events and contraindication in COPD. And so my question is there an experience from Exubera with regard to adherence with doing pulmonary function tests before the actual prescription of the drug? I know that falls outside of the FUSE data and it may not be available, but one of the issues has always been that there are many patients, some of whom have actually never smoked, who have chronic obstructive pulmonary disease, who may be unaware of that, who are therefore at risk for the adverse events of an administered insulin agent, whether Exubera or Afrezza, that are unrecognized. And the premise of pulmonary function testing before requires some adherence. So my question is, what do we know about that adherence?
Mia Paternity, FDA. We did inquire about some of that data, but the sponsor was unable to provide that for us. We are recommending, as for Exubera, for the same for Afrezza in terms of the pulmonary screening, but you bring up an excellent point.
Yes.
Hi, this is Banu Karimi Shah. I'm with FDA. I think it's also important to remember that with Exubera, it was labeled and recommended in the labeling to measure the pulmonary function test before. It wasn't a requirement, so we didn't, from FDA's standpoint, it wasn't tracked like it might be in a risk management strategy. So for that reason, we don't really have that data.
So are you suggesting that if, as the drug and labeling is proposed here, that it in fact would be tracked prospectively?
The way that it's here is it's a labeling recommendation.
I see.
So of the people who did not get an opportunity to ask a question but wanted to this morning of the sponsor, I don't want to go to those questions for reason of the schedule, but if any of those questions would you anticipate requiring the sponsor to do some work to pull together some information, so they might bring that back to us, I think we should hear about that now. Is that the case for anyone? Dr. Soler.
So many of my questions to the sponsor regard the issue of spirometric quality. I think that we've established that it's a reproducible test, but it does require quality control. And we can come back later to the quality control blinded independent review, some of the details of that. But one of the questions emerges performance of spirometry in American centers versus non-American centers. And I'd be interested in seeing the data about FEV1 decline stratified by the 40% of patients from at least 175, I believe, or 171 rather, that were U.S. versus non-U.S. And similarly, the sponsor showed the data about 030, where we actually don't have, at least I didn't find the prevalence of U.S. versus non-U.S. I'd be interested in seeing the FEV1 decline data stratified by center, looking for center effect with regard to quality of pulmonary function testing.
Okay, so if the sponsor could look to bring that back later this afternoon. So we're now going to take a break for lunch. At this point, you should please take any personal belongings you may want with you. The room will be secured during the lunch break. Panel members, please remember there should be no discussion of the meeting topic during lunch among